innovative ideas and a shared responsibility towards our planet. In a world where climate change is no longer a distant threat but a lived reality, agriculture stands at a crucial crossroads. The need of the hour is resilience. The need of the hour is innovation. And above all, the need of the hour is youth-led transformation. This summit brings together brilliant student researchers, future policymakers, agri-tech innovators, and sustainability champions, all united by one purpose, to reimagine agriculture that is climate-ready, technology-driven, and food secure.
Once again, I warmly welcome you all to this inspiring summit. It is my privilege to invite Dr. Tulsi V, Professor and Head of Department of Soil Science, RARS Pattambi, to deliver the welcome address. Ma'am, we are honored by your presence. Kindly address the gathering.
Good afternoon, everybody. Be the volcanic eruptions of Mauna Loa, forest fires of Australia, steaming chimneys of Manchester, or the poison-spewing vehicles on our Kerala roads. All these have one thing in common, that is they emit carbon dioxide into the atmosphere. This much miracle molecule, the carbon dioxide, along with other greenhouse gases, is making the much debated and discussed topic of climate change a reality, affecting the agriculture scenario and food security. In this context, Kerala Agriculture University is organizing a 2-day student summit on climate-ready agriculture, the innovations and agri-tech solutions for food security, under 2 themes as discussed as detailed in the brochure. And today is the first session of the program under theme 1, and we are very delighted to announce that we have got more than 100 papers for presentation in this program, and today we are blessed with the presence of Dr. Prakashan Chelladan Veetil, the agricultural economist of International Rice Research Institute. Dr. Prakashan is a senior scientist at the International Rice Research Institute, specializing in agricultural and behavioral economics. With a PhD from Gen University, Belgium, he leads major initiatives across South Asia focused on transforming agri-food systems, advancing climate-smart agriculture, and improving farmer livelihoods. His work includes developing innovative crop insurance models, strengthening value chains, and promoting rural entrepreneurship. Dr. Prakashan has led key components of CGIAR programs and contributed to policy-oriented research on technology adoption and socio-economic impact. He has published widely in international journals and is an active member of global agricultural economics associations. Sir, we welcome you to this program, and your presence, mere presence here itself is an inspiration for the generations to come. We welcome you, sir, for the program. Let me now take this opportunity to welcome Dr. Lada A., the Director of Students' Welfare and Coordinator of the Kera Cell of Kerala Agriculture University. Ma'am, your vision, your expertise, and your leadership have been instrumental in shaping the activities of the Kera Cell as well as Kerala Agriculture University. We are honored to have you here, ma'am. Welcome to the program. Let me take the opportunity to welcome the eminent faculty present here, Dr. Sudhir KP, the National Fellow and Head Agri-Business Incubators, and all other dignitaries present in the audience. May I now take this opportunity to welcome the judges or the panel of experts who will be arriving shortly, Dr. Veena, Dr. Gavas, Dr. Smita KP. Their expertise and time is greatly appreciated. And most importantly, may I take this opportunity to welcome the student delegates of this program who have taken pain for applying for the program and have come for oral presentation, and you are the stars of this particular program, and wish you have a great day and great presentation, great experience ahead. And as we all know, teamwork makes dream work. There is a team behind this program, and may I now take this opportunity to welcome all my colleagues of the Kera AWD team, all faculty present here, and all others who have gathered here for making the program rich and lively. Let me take this opportunity to welcome one and each one of you present here to this program. Wish you all have a great experience. Thank you.
Thank you, ma'am. Research gives direction to agriculture and policy gives it strength and sustainability. In the journey towards building climate resilient farming systems, it is essential to connect the realities of the farm with the decisions made at the policy level. With that thought, we now move to the most awaited segment of today's session, the lead talk. It is my proud privilege to introduce our keynote speaker, Dr. Pragashan Chelladan Vittil, agriculture economist at the International Rice Research Institute. He will be addressing us on the highly relevant theme, climate resilient agriculture, farm to policy. Sir, we are truly honored by your presence and look forward to gaining valuable insights from your experience and expertise. With great respect and admiration, I invite Dr. Pragashan Chelladan Vittil to kindly deliver the lead talk.
Okay, good afternoon to all. Thank you, Dr. Tulasi, for very good introduction. Very nice introduction. I thought I can skip a couple of slides because of your introduction to mountains and volcano eruptions and flooding and whatsoever it is. So, it's good to discuss about, especially in the student summit, it's good to discuss about a topic that we are all passionate about, and it's affecting each one of us. Why it's affecting? Probably there could be some issues happening here. If you look at the miracle molecule that Tulasi talked about, the carbon dioxide, what is happening in the last 4 decades? So it's showing, the red dot showing the observatory is from the NASA about the carbon dioxide emissions. It's emitting through the fossil fuels or other human activities that including a lot of burnings, agricultural land use changes, etc., etc. Every year we are emitting now more than 36 billion tons of carbon dioxide to the air, causing significant climate change effects that including the erratic monsoons, the heat stress, the whatsoever we can name it under that. This graph really showed over the last couple of decades how the carbon dioxide emissions are increasing, and in 2013 it crossed the mark of around 400 ppm level in the atmosphere. That's happened, that never been a phenomenon in the last 8 lakh years in the history of Earth. So we are crossing the limit. Are we going to change this or arrest this? There are IPCC already highlighted about different scenarios as well. And definitely the anthropometric CO2, that's man-made CO2s are really, really huge. And this concentrated mostly in the developed nations, but it's spreading to larger economies like India, China, etc. So this graph shows the net emissions. So there is, as I said, the due to fossil fuels or other land use changes, etc. There are several emissions happening. That's surface emissions. And also there are removals, that's the sinks, that is through the absorption of carbon dioxide. For example, the plants, when it produces the crop, or like certain types of rocks are weathering, then that also absorbs the atmospheric carbon dioxide and lock it in the land, either as an organic matter, carbon, or some other forms of carbon. So it can change the land carbon stock in different countries. So some in this figure there are some are shown with. Okay, I think I cannot use the pointers. So some are. Let me try if I can. So some are green. That means they are net removing countries in terms of their carbon budgeting. But some are like dark brown or light brown colors, they are actually emitting more. So the countries like China, USA, India, Brazil, and Southeast Asian countries, they are bigger emitters. So this changes how this combined effect will have a lot of stock changes in the land, the agricultural land, for example, or in the forest land, and this will have impacted how a land use is happening and how the land use changes are happening in the world, and it is drastically changing. When if you go to the Western Ghat 50 years ago and now, you could observe, or if you are traveling from north to south, you could see the land use changes significantly. The land patterns, the cropping patterns, the land uses, it's all changing throughout India because of the fast developmental process. Then the effect of those climate change is like temperature increases, or like seasonal erratic monsoons, or heat stress, prolonged extreme weather events, any such extreme events. This will have an impact on the food production. But until now, the pace of food production, the scale, the growth of food production, it outgrows the population demand. So currently, but this will have significant impact in terms of the productivity growth. For example, that's more of in the tropical climate, like specifically in India, African countries, and many other countries. It will have a significant impact in terms of the total factor productivity. What does it mean? We are tending to overuse resources, whether it is water, whether it is fertilizers, or many others, without yielding properly. So this serious impact, it will have consequences on the incomes as well as the livelihood of millions of farmers. Take an example of India's total factor productivity. In the early 2000, it is somewhat 1.9. If anyone is familiar with that numbers, you may understand it a little bit more. But translating in 2000, it is 1.9, and 2018-20, it is somewhat 1.1. What does it mean? There is a significant change in terms of the sustainable, in terms of use of inputs in sustainable manner. So this growth gap is an important concern for the agriculture, and especially in developing countries. And what would be that cost? It would be on an average, the global estimated cost is somewhat $650 billion US. So it's a significant impact. And out of that, around 300 billion is specifically with land degradation, like changing the pattern, the soil, and also the land use patterns changes, etc. And around 140 is extreme weather events, and remaining is mostly the cost of pest and diseases. So we tried, it is becoming more and more use of pesticides and other measures into that. So what happens in this case, like we have what happened after the post-green revolution, or green revolution and post-green revolution era, as it is shown in the left, this particular evolution map. It's showing the water, expanding in the irrigation, and below down it's showing how the groundwater stress is happening. So right now we are in a situation wherein India's aquifer, like on an average, if you look at aquifers, are dangerously depleting. So 60% of the districts, 60% of the total districts are under critical or over-exploited conditions of groundwater. And out of which, more than 90% of the groundwater use is explicitly for irrigation. What does it mean? It means this growth, this issue will have consequences on the productivity in future as well as our livelihood, as well as the drinking water issues, etc. So, so it will also have this climate change, will also have impact on the whether the agricultural laborers who are working in the field. So let's see. We have two emission scenarios, moderate emissions and high emissions. High emissions means like we don't do things business as usual, the current way it is moving. In this case, we observe like in several parts of the world, increasingly the number of the laborers who are exposed to heat, 103 degree Fahrenheit, or it's like 39.4 degrees Celsius, more than that, it's increasingly higher, becoming higher in many parts of the world. So uh, this is one effect. Another effect is the farmers or the poor people, their climate vulnerability is extremely becoming a concern, specifically in Africa and Indian conditions. So where there are predominance of smallholder, more than 70-80% of the land holdings are smallholders. That means it's not just the vulnerability. So what does it mean by that vulnerability? Their adaptation capacity, climate resilient adaptation capacity will be highly limited, and it will in turn lead these smallholder farmers into a vulnerable cyclical vulnerability problems and into a poverty trap. And it will also have impact on yield. So I'm showing here two yield predictions from the NASA's models. All these are NASA models, many of them. Here it is the maize. If you really look at the maize is predominantly grown in tropical equatorial regions, some of these regions, and wheat is growing in temperate regions in the right panel. So maize yield on an average is going to decrease by end of this century is around 25%. But not the world is doomed, but wheat yield is going to increase by more than 10 to 14%. Assumptions, modeling, all those has to be acknowledged. What does it mean? The wheat is a temperate crop. The area that can be cultivated by the wheat will increase, but the tropical part where we live will have a significant impact. Unfortunately, most developed countries are in the temperate region, so the millions or billions of people live in this region is going to suffer a lot. And it's not just wheat or maize, but also for rice. I'm giving three conditions, three scenarios, RCP scenarios. One is a high mitigation, 2.6, and 8.5 is like business as usual. That's a high emission scenario. So this is low, very low emission. This is high emission scenario. This is moderate emission scenario. In that conditions, in business as usual, the total rice yield will significantly impact more than 15-20% in high, if we continue the current situation, that will reduce. But if it is a low emission scenario, extreme tight situation, that means we kind of follow the Paris agreement, 2 degrees Celsius by end of the century, then it would be within the limit. I'm mainly focusing on Asia because that's a rice bowl of the world. So rice production is very critical in global food security, but there are natural hazards, right? And how this could be minimizing. So I'm just highlighting a couple of climate resilient, low emission technologies. So to highlight, one, residue management, short duration variety, water fertilizer management, specifically in that case. So on an average, it can reduce emissions, primarily methane, by 65%. So it varies with different, and that's our experience from here also in Kerala conditions. So water management could be alternate wetting drying, mid-season drying, and also micro irrigations, etc., etc. Residue management, it can happen in situ or off field, and nutrient like slow releasing fertilizers, site specific fertilizers, and varieties, climate resilient varieties, short duration, low methane emitting varieties, and then mechanized uniform sowing, emergence using mechanized precision farming. So look at the variety. So there could be accelerated breeding for climate resilience, right? And this could be facilitated in olden times, like even before Mendelian times. The civilization happens with domestication of agriculture. So there is a selection process, a breeding, a primitive breeding aspects are happening in that, and then it comes to Mendelian hybridization, artificial manual selections, etc., etc., looking at the phenotype, looking at the yield, etc., etc. And most important, this design is moving from that traditional era to a smart breeding design. So what does it mean? It's comprises of like use of different types of molecular genetics that's evolved during 1990s, early 2000, that period, to the latest, like including gene editing, etc., to the latest technologies that include the artificial intelligence for example, like the genome selections, panel selections, to smart designing of the varieties, etc. So this is very interesting case, and China is transitioning to the stage 3. So this is the stage 1, stage 2, and this is stage 3, where we are supposed to. It's better, it's always important as a researcher, being a student, being a faculty, being a university leadership, what do you think where we are, our university, and also the government, where we are actually aiming at. So this is actually something we need to really prioritize for breeding. And some of those things, like, okay, even in IRI, we have our International Rice Research Institution, we have the South Asia Regional Center at Varanasi. We started some of those information to be in practical use. That include the genome laboratories, use of AI in selection, identifying genes, etc., speed breeding facilities, and computational biology phenomics into it. So it's not just one. In that context, we should be using different types of data that include the remote sensing data, the sensing data, like the way we use water sensors or other sensing data, and then the genome sequencing data and other meteorological data, etc., etc. So, and we also have witnessed a paradigm shift in genome editing, the policy shifts. Earlier it was coming under there was a confusion and coming under genetically stringent GMO category, genetically modified organisms regulations. But when it comes to two types of editing, that's within the same genome sequencing, SDN1 and SDN2, that's not really coming under the GA, the Genetic Engineering Appraisal Committee regulation, GMO regulations. That's a 2022 policy. That's relaxation. But still, if there is larger gene insertion, still coming under this. So what do I mean by this? Like the AI summit is happening now, and that will also hopefully create a lot of opportunities for this type of holistic changes, breeding, and then policies, etc. And we need to ensure such development is happening. Second, climate resilient is adjusting water management from current traditional practices of continuous flooding to more of mid-season drainages, alternate wetting and drying. So I said total factor productivity is less, so in this context, it could also increase, and also the emissions through the reduced methane emissions here. So, but it's not enough, because this is everyone knowing this, what is new in that, and how we translate that into modern applications and tools. That's what we have to use, IoTs and AI systems. To give an example here, we have installed, even if you go to Manuti, ARS Manuti, the rice fields, you can see the sensors here. and it is connecting to the gateways and providing information when to irrigate. So it's optimizing, using intelligence to optimize. We are using analytics. Right now, I give an example of Bihar here, and there is the southern Bihar, these green patches. It's having the highest electricity or investment for electricity and irrigation investments. So, but the yield responses says additional investment will have significant gain in northern part here, the green patches here. So there is misaligned investment, and that's an important dimensions to look at here. And soil, we do have a lot of designs here, for example, predictive soils and soil maps, where which fertilizer can go where or which nutrient can go where, etc., and which is connected to, for example, this is the Andhra Pradesh has initiated a policy on subsidizing zinc fertilizer, but it doesn't give any significant impact. Why? Because it's like a uniform support program, and it doesn't really target the needy farmer, so it doesn't show significant effects. But also, like, we have been testing, and it's very important to see that the slow releasing, for example, urea briquettes, etc. I think that's one of the topics in Kera. Also, we are trying the PhD topics to explore into slow releasing ureas, etc. Then bio inputs that include the biochars, for example, that stocks the atmospheric carbon into long term, thousands of years into biochars, and then not thousands, I think hundreds of years or thousands, yeah. And then reduces the greenhouse gas, the methane emission as well, as per our initial trials. So we need more verification around that. Then the organisms. There are new generation biology materials, microorganisms, methanotropes, an example, and there are several like growth stimulating, resilient stimulating microorganisms that would be very helpful. And then fertilizers like sulfur containing or phosphogypsum here is showing a significant impact in terms of methane reduction. And more important here is like talking about how we design a low methane emitting rice varieties for the state. And we did conduct trials in different parts of India. On an average, it shows like alternate wetting and drying reduces the emissions by more than 60%, around 50% or more. And also the water irrigation saving by 32%. Even in case of rain-fed rice systems, just a direct seeded rice will decrease methane 10%. And also there are different effects here. And most important is the irrigation water savings. And if you recall one of the panels, I was discussing about groundwater stress, and this will be a huge relief for many groundwater stress or in case of here surface water. And there are a couple of policy dimensions here. So if you really look at India's budget, the ICR budget this year has reduced from the last year budget. So even though the research investment is significantly reducing all over the world. This graph shows one means the intensity is 100%. Look at our countries, low, middle income countries. We are investing less than 50% of what we really needed. The developed countries are investing higher. It's increasing more and more for the climate smart agriculture. So in this case, what we are ending up, the gap is widening, and the competitiveness of Indian agriculture or this developing country agriculture is very, very weak. And where we will be ending up, a reduced, the higher poverty rate, not targeted to be here. So on an average, the investment gap is 15.2 billion. That's what estimated for climate smart in that. And what would be the global global subsidy, 700 billion. So it's just a fraction of that we'll be investing, will have a huge impact in the global scenario here, reducing the poverty and improving the livelihood. And sometimes the impact, the policy over mostly like look at, oh yeah, it could be bundled with some social technical bundling. So that's an option. Say we had a bundling for crop insurance with agricultural loan. It has been changed recently. So we tested in Orissa over the period of 2018 to 22, randomized control trials in around 300, 160 villages across Orissa, and we were looking at what would be the short-term and long-term impacts. For example, this is a crop insurance, and we see only marginal increase for, we saw very significant increase here, that's in the short term. But in long term, it is plateaued. And it's the same case here, the seeds, stress-tolerant rice variety. We don't see a long-term significant impact of such bundling in this case. So it is, I'm not suggesting not to bundle. Yes, there is a bundle, but we need to be careful what to bundle and where to bundle. So some of those are research interests for the students here, like integrated research. And this is hopefully my last slide, and how the partnership for climate resilient agriculture, where do we need to invest? In CARA, we are trying to explore this in a way wherein we will have a network, a partnership built on academic research as well as the scaling or the use case or industry participation. So how does it look like? We have the, because in CARA we are focusing mostly on the water management, emission reductions, and climate resilience. So we are looking at field measurements, different types of low emission agriculture sectors, and also introducing climate models into there. But at the other side, it is more of using advanced tool, like the way I was talking about breeding, how can we use for agronomy research here. And this is a typical example, probably we will explore and see. And there are building blocks we tried with, we are working on climate models, we are working on PhD networks, we have field experiments, sensors, etc., and also the latest NISAR satellite data also we would be using. I hope today, tomorrow, this section, climate resilient agriculture, will be very useful, specifically with students. I have one last thing to say. MIT, Harvard, IITs are not famous for its faculties. Never think that it is meant for faculties. It's famous for students, because of students. The original creativity, ideas start from you, the students. So I hope this type of events will facilitate such innovations to come. Thank you all. Thank you for the opportunity to talk here.
Thank you, sir, for the insightful and enriching session. As a token of our gratitude and appreciation, we would like to present a memento to you. May I request Dr. A. Lada, Director of Students Welfare KU and Convener of Student Summit 26, to kindly come forward and present the memento. Let us express our heartfelt appreciation with a big round of applause.
We now move on to the oral presentation session. Each participant will be given a total of 5 minutes. A warning bell will be given at 3 minutes, and to stop the presentation, a long bell will be at 3.5 minutes. A bell for alerting the judges to end the discussion at last 1.5 minutes. I repeat, a warning bell will be given at 3 minutes. To stop the presentation, a long bell will be there at 3.5 minutes. A bell for alerting the judges to end the discussion at last the 5 minutes. Let's start the presentation. OPA 05. Judges, please note OPA 05. I kindly request the judges to be please seated.
Today's refreshment is sponsored by Agri Business Incubator.
First call for OPA 05.
Good afternoon, all. I am Meenakshi Kailas from the Department of Agriculture, from the Department of Agronomy, College of Agriculture, Velayani. Today, I am here to present a paper on the topic Big Pro, a weather-based crop simulation model for optimizing sowing dates and nutrient management in yard-long bean. So the objective of the experiment was to develop a growth simulation model for yard-long bean to determine the best planting time, the potential yield gap, agro input required to reduce the yield gap. So these are the details regarding the design of the experiment. The crop was sown on the first day of each month, starting from October 2024 to February 2025. And these are the details regarding the treatment. The main plots consist of 5 different sowing dates, and the subplots are 3 different varieties of cowpea. And these are the computed parameters recorded during the experiment. And these are the weather parameters which were recorded daily till the final harvest. And these are the growth attributes recorded at 15 days interval. And these are regarding the yield attributes. And the methodology for developing the model. So the structure of the model, it consists of driving variables which are 9 each of weather and plant parameters. And this is the system diagram of the model, which is, so system diagram of a model actually represent the way in which the different components actually interact with each other to produce a given output. So here, as you can see, the GDD, that is growing degree days, was used as a major parameter. And here, the growth phases of the cowpea, it was divided into 5 different phases, starting from sowing to the final harvest. And 3 months were chosen for developing the model, that is October, January, and February. And this is the consolidated table which was used model Big Pro. Now, the salient findings under field experiment, it was found that the treatment combination D5V2, that is sowing of Geetika variety in February, was the best performing combination, matched with that of the real field observations. So, and NOF actually represents the degree of mismatch between the model prediction and the observation. So, here NOF, if it is nearing 0, then it means good model prediction, and D index nearing to 1 means good model predictions. So, here you can see that, and in the graphs, the blue lines represents the observed value and the orange lines represent the predicted model predicted values, and it was evaluated for different parameters like leaf number, number of branches, leaf area index, dry matter of the plant, dry matter of stem, etc. The model also predicted the potential yield for the 3 different varieties with an optimum temperature of 30 degrees Celsius, and the maximum potential yield was recorded for V2 followed by the V3, that is KU Deepika, and the least was recorded for Valayani Jyotika. Now regarding the yield gap was least for the December zone varieties, and it was highest for the December zone variety, and it was least for D5, that is February zone. Similar trend was observed for other varieties also, that is Valayani Jyotika and KU Deepika too. Then regarding the percentage accuracy of the model, these also showed the similar trend where it was highest, the accuracy was highest under February and the least under December zone crop. Now regarding the agro advisories developed by the model, so here you can see the amount of NPK requirement which was predicted by the model. So in the table you can see that the NPK, this actually variety had the best, had a higher NPK requirement compared to the POP recommendation. Now to conclude, the model validation showed that the D index value near to 0.99 and also NOF value as low as 0.02, meaning that the model has good model predictions, and the potential yield simulations indicated that V2 variety, that is Geetika, was performing best, followed by variety KU Deepika, and the best performing treatment combination was Geetika shown in February. And so the field observation actually matched with that of the model predictions, meaning that the model was accurate, and the model also highlighted the need for a variety and state specific nutrient requirement to achieve the potential yield. Thank you.
So you have calibrated the model, or you have developed?
We have developed a new model, ma'am.
Developed a new model,
yeah,
based on a single season data?
Ma'am?
Based on a single crop data?
Yeah, actually we have sown the crop for 5 different months, and we have taken the field observations. Like, so these are the different weather, parameters that we have calculated under field conditions. Like length of the vine. So we have calculated these under field conditions, and we have divided that according to the, we have calculated that for according to GDD. So GDD has a, that is growing degree days, it has an equation. So we have we have calculated for each of the parameters, we have calculated the values for each of the parameter, and we have developed the input data as given in the file. So these are the
So your input data are meteorological parameters, and the output is?
Output is what the model has predicted. Like based on the input parameters we have fed, the model will predict the how will be the growth of the model.
Okay, your language used is which?
Python code.
Python language.
Python language.
Okay, thank you.
So we'll move to the next presentation.
Next participant, judges please note OPA01. I repeat, OPA01.
Presentation ചെയ്താൽ പോരെ ഇത് സ്ലൈഡ് ഇതല്ലേ ഫോർവേഡ് ഓക്കെ യെസ് സൈഡ് Good afternoon, and my name is Thangachan, and I am a research scholar from Rajagiri College of Social Sciences, Kalamashiri. And my topic is bio-high-tech mushroom farm. We have been in the mushroom domain for the last 18 years, and this is a case study of our experience during the last 18 years. We faced a lot of problems and lot of challenges during our 18 years time, and among all the problems, the climate variance and pest, fungal, rodent attacks are the severe ones. So this is a conventional farm which is susceptible to this climate variance, rodent attacks, and all these things. And so we made some creativity in the mushroom farm and came out a climate resilient bio-high-tech farm to address these issues. The durability of the farm was yet another issue. So all these things were addressed with our creative model, which you can see on the other side. That is with an air gap system on the top and side, then white metallic cladding sheet system to reflect the heat, then complete insulation of the inside of the farm, which we could not show in this 2D picture, with a double-sided aluminum bubble sheet system, then ventilation system, double door system, etc. So all these things address the above issues. So going back, the bio-high-tech farm has a life of 30 years as against the 5-6 years of the conventional one, or with the black and white sheet system which we are using in the polyhouse. So with this innovations, we could achieve a 6 times increased productivity and less than 5% pest and fungal attack as against the 20 to 30%. So, as we can see in the graph, during the summer, even though we are using that fan and pad system with our creativity, it is a proactive to the climate again. So monsoon we get 120 to 130 percent production high, where in summer it was a collapse earlier with the conventional farms, but we could achieve 80 to 90% production even during the summer. So we can see the 1,500 kg, that the life of this farm has increased to 30 years with that metallic sheet system as against the black and white sheet system. Then the annual production has increased from 1,500 to 6,400 kg, that is 4 times yield. Then revenue also increased to 506 increase is seen, as we can see in the graph. So these are things which again corroborates our shift from the conventional system to the fan and pad system with our creativity. And we got that institutional validation, and recently we got a contract order from the government agriculture department to do this farming at Mavelikara district farm system. And so we can say this is a validated and scalable and profitable model for the farmers to follow. Thank you.
Hello, sir. Regarding this, this is an engineering innovation you are saying.
Yeah.
Have you done it already, practiced it somewhere?
Yes. I think we have done it in our own premises, and in Kerala we have done about 20 such models.
So you have said
and
about...
the last one being done, it is continuing at Mavelikara district agricultural farm.
Okay. So you are saying like the contamination rate has gone down.
Yes.
Is it how you have arrived at this from all these cases wherein you have erected this, or is it from one model?
We have a model.
Is it a case? I'm asking, is it a case study?
This is a case study, but this model has been implemented and we have been implementing it all over Kerala.
Okay. And what is the initial cost of this?
This 500 square feet, that farm will cost you 4,95,000 rupees, and which we can recover the capital investment, which we can recover in the first year itself.
So you have calculated the economics of it?
Yes.
Like how many years it will take for the payback?
Yeah. In 1 year, we will get back that capital investment.
But we have not seen it, sir. That's why we have asked.
I have shown it in that.
Economics
so because of the time constraint, I have to move the slides again and again and again.
Yeah, okay. It's fine.
Okay, thank you. Thank you for the great presentation.
Thank you.
The next participant, OPA02. OPA02.
So good afternoon to all the respected dignitaries, staffs and student friends present here. Myself Sneha from second PhD doctoral scholar from Tamil Nadu Agriculture University, Coimbatore. I am going to present my topic of evaluating the impact of flood inundation on rice growth in coastal regions using Sentinel-1A satellite data. Actually here, the objectives of my study is to delineate the flood-prone areas, to map the flood-vulnerable areas, and to study the impact of flood on rice crop. As we all know, climate change is now a rapid thing which is decorating ourselves in the forthcoming climate change and environmental conditions. So, as for to control and to mitigate that, we have to control the flooding rate also. So, coming to here, my study area is in coastal regions of Tamil Nadu, namely Nagapattinam, Mayiladuthurai, Kadalur and Thiruvarur districts. In these areas, we have mapped the flood areas for continuously for past 7 years, right from the year 2015 to 2022. Here the methodology which I was used here is, I have downloaded satellite data from Sentinel-1A and we have calculated maps through the satellite data, and through this I have gone for ground truth survey to the coastal regions also. Then by combining both the maps, I have calculated the rice area maps and flood area maps. Then by combining both the maps, we have assessed the result here. Here I have the softwares like ArcGIS, QGIS, MS Excel, Mapscape Rice. Mapscape Rice is for mapping the area of rice, and ArcGIS and QGIS for downloading and processing of satellite data. The methodology here used is for flood area mapping. First, acquisition of data from Copernicus Satellite Hub, which is from Google website. And second is pre-processing of satellite data. Third is pairing of the images. Fourth is registration of the images through satellite data. And the fifth is changing the detection of the images just to find the area which is properly classified. And the sixth is thresholding and classification. Seventh is validation and accuracy assessment. And eighth is visualizing and reporting of results. And next is rice area estimation. This is which I have done through the Mapscape Rice software. Overall, the results which I have got is right from 15 to 22. In 2017 and in 2018, no floods have been observed in those coastal regions. In 2015, the higher area which is observed is severely 58,336 hectares in 2022, and it was less in 2019, which is about 4,133 hectares. These are the flood-prone areas which I have shown in the previous slide. This is for 2015. Same for 2019, 20, 21 and 22. So combining all these, we have observed major flood affected area in the year 2022. Then this is accuracy assessment. I have gotten a kappa index of 0.72, which is a high defining accuracy. And overall accuracy here was 86.2%, which is a very good accuracy. This is the confusion matrix. Through this, I have calculated all the producer accuracy, easer accuracy and overall accuracy. And here is flood inundation and risk associated areas. This I have calculated for finding out the continuous flooding regions in those coastal regions and to alert the farmers based on the flooding availability. And next is rice area estimation, which is done through Mapscape software. For all my coastal regions, Kadalur overall rice area is 1,347 hectares, and for Mayiladuthurai 52,806 hectares, and for Nagapattinam 47,473 hectares, and for Thiruvarur 1,77,834 hectares. This is impact of flood and rice. This is the very important result here which is obtaining here. First we have to calculate the flood area. Second we have to calculate the rice area. Then by overlaying both the areas of flood area and rice area, based on the extent of overlap, we can identify the severity of damage here. Say for example, if the overlap is moderate, then the severity is higher here. If the overlap is significant, then there is moderate damage. If the overlap is less, then there is very less severity. These are the maps I have obtained so far. These maps are obtained using ArcGIS software. So, how we can mitigate is that we can give early warning systems, crop insurance or financial support affected, improve the drainage systems, we can develop some flood resistant varieties, we can improve the better land management systems. This is the future line of the work. Thank you.
It's okay, your work is good. Can you predict, with this model, can you predict the flood in the previous years or coming years also?
Yes, ma'am.
How accurate? How much accuracy will be there?
Actually, accuracy will be more than 80%. It is based on the rainfall data. If the rainfall is more rainfall prone zone areas, we can calculate the severity based on the amount of rainfall occurring there.
Did you validate it with other parts of Tamil Nadu?
No, ma'am. I have not yet validated because my study area is those 4 districts.
Okay, thank you.
The next participant, OPA03. Judges, please note OPA03.
afternoon all. I'm Suman George and I'm going to present a study on the topic pigment profiling of selected fruits and vegetables and their role in climate-ready nutrition and food system resilience. Apart from coloration, the plant pigments such as chlorophylls, carotenoids, anthocyanins have an important role in physiological process of plants as well as in the human nutrition. The objectives of this study is to quantitatively estimate the chlorophyll A, chlorophyll B, carotenoids and anthocyanin in selected vegetables, fruits using spectrophotometric method to compare the pigment composition and to identify the pigment-rich food sources. The materials selected for this study were the red and green varieties of amaranthus, red and green varieties of cabbage, 3 color varieties of capsicum, and 3 varieties of chili, and 4 varieties of watermelon. For the estimation of chlorophylls and carotenoids, the plant tissues were homogenized with 80% acetone, and the supernatant after centrifugation is used for obtaining the absorbance at 663, 645, and 470 nanometer using a spectrophotometer, and the values were calculated using the formula. For the estimation of anthocyanin, pH differential method were used in which the plant tissue is homogenized with acidified ethanol and the supernatant is mixed with the 2 different type of buffers, potassium chloride buffer and sodium acetate buffer. And the absorbance were measured at 520 and 700 nanometer, and the absorbance were used for calculating the values with the formula. We're comparing the amaranthus varieties. The green variety has more chlorophylls, while the red variety has more anthocyanin. When comparing the cabbage varieties, the chlorophyll is more in green and anthocyanin is remarkably more in red variety. When comparing the capsicum varieties, the chlorophyll A and B is more in green variety and carotenoids were more in red and yellow type, in which the red has more amount of carotenoids. When comparing the chili varieties, the long green type chili has the highest chlorophyll contents and the least in the Hungarian wax chili. When comparing the watermelon varieties, the carotenoids level were more, in which the Kiran type of watermelon has the highest carotenoid content. So when we are comparing the all food items together, the green and red amaranthus has the highest chlorophyll A content, and the green and red amaranthus has high chlorophyll B content, and the carotenoid contents were more in red and yellow capsicum and in the watermelon varieties. And the amount of anthocyanin is more in red cabbage and red amaranthus. So these functional pigments has a role in the climate-ready nutrition, in which they act as they perform different functions in the physiological process of plants, in which the chlorophyll-rich vegetables have high will perform photosynthesis. The carotenoids have antioxidant capability, which protects them from environmental stresses, and the anthocyanin, which protect them from UV radiation and oxidative stress. And the different food color will contribute to different health benefits, in which the different colored food has different pigments which performs different functions in our body. So actually the pigment-rich crops supports dual resilience, the plant resilience and the human health resilience. So the pigment diversity in fruits and vegetables strengthens the climate resilient agriculture by enhancing both plant stress tolerance and the human nutrition security. Thank you.
Have you analyzed this as your work?
Yes.
Or is it a compilation of all the from various references?
No, the values were taken by me.
Because I am doubtful, because all these vegetables has already been assessed for all these things.
Yes.
Then what is the novelty in your work?
So, my contribution is the published work is almost related to my data.
The varieties are different, no? You
Yes,
have
yes.
selected some special varieties and you have analyzed.
Yes,
Is
yes,
it so?
Yes, yes.
Okay.
Yes, one minute. What do you mean by this climate-ready nutrition? What do you mean by that? Climate-ready nutrition.
So means the the functional pigments act as the different functions. So for a climate-ready nutrition, we can use the type of plants with a particular more pigments for a particular space or a particular geotropical area.
What is the take-home message of your work?
My work means they calculated some varieties which I get from market in which available in different color varieties. So I selected randomly and tested the pigment concentration.
Okay.
Is there any nutritional difference between this red cabbage and green cabbage, as well as these three colors of capsicum? Nutritional content.
Means the pigments.
Yeah, based on the pigment, it's nutritional content relation.
Means the chlorophyll is more in green, but the level of anthracene is remarkably high in the red. So it's a good antioxidant providing food, the red cabbage.
Did you analyze those antioxidants? Did you estimate it?
No, I estimate the pigments only.
So, assumption only?
Yes. From the previous earlier literature, I get the data.
Okay, thank you.
Thank you.
The next participant, OPA04. OPA04. A very warm good afternoon, respected dignitaries and all others who have seated here. So, around 1.8 billion people in the global level has been affected with the drought and the climate vagaries. Considering this in mind, we have conducted the research work on drought management in rice using soil amendments. The primary objective of the study is to assess the impact of drought mitigation strategies on anatomical, morphological, biochemical profile of rice, as well as how it is impacting the productivity of rice. So these are the methodology, and here we have followed factorial CRD as the experiment, and there were 5 soil amendments and 3 moisture stress levels. And the first amendment is silica, which is fine silica of potassium silicate, around 100 kilogram per hectare was the rate. And biochar, which is made out of rice husk, it was applied at the rate of 5 tons per hectare. Then Pusa hydrogel specifically was taken for the hydrogel, and 25% of additional potassium was for treatment, and control a check without any amendments was also given. And 3 moisture stress levels were, first one, that is 50% field capacity, reducing the levels 50% than what is required for the rice. Then stress, which was given at maximum tillering and panicle initiation stage by withdrawing water 5 days prior to maximum tillering as well as panicle initiation stage. Then giving water when about 90% of the plant shown wilting symptoms. Then no moisture stress condition, giving water as much as rice requires. So medium duration variety Uma was taken for the study, and the season of planting was late Rabi season. So these are some of the pictures of the rice husk biochar. This is the fine silica that is applied, and this is specific Pusa hydrogel, and this is the condition when it absorbs water, and this is the leaf rolling symptom, after which the water was given in the second stage of irrigation. Moving on with the results. So first one, the effect of the soil amendments and moisture stress on plant height. We could see that at 30 days, under irrigated condition, potassium application had found the highest plant height, but under 2 stressed conditions, we could see that silica showing the taller plants. A similar trend was also observed in the 60 days of planting too. Under no moisture stress irrigation, the potassium was showing taller plants compared to the other treatments, and the stressed condition, still the silica as well as biochar was showing taller plants. Again, under 90 days, the similar trend as of 30 and 60 was seen. Moving on with the dry matter production, again, in silica, we could see that a highest plant height was, highest dry matter production was seen in maximum tillering as well as panicle initiation drought stages. And when compared to this irrigation condition, it was again biochar followed by the silica. A similar observation was also found in when the dry matter production was observed at panicle initiation stage. However, there was no much difference in treatments when at the harvest. Moving on with the yield parameters, we could see that the number of grains per panicle
was highest obtained under irrigation conditions from the 25% of additional potash. However, when it was stressed condition, similar to the growth parameters that we have seen, the silica found highest number of grains, which was followed by the biochar. And similar in the cases of 1000 grain weight also, we could see that highest 1000 grain weight was observed in 25% of additional potassium recommendation, which was followed by silica under irrigated conditions. Coming towards the stressed conditions, it was biochar followed by the silica. Moving on with the important parameters, that is grain yield. So this green line represents the 50% FC conditions. We could see that the 40.9 grams per plant grain yield was obtained when the silica was applied, and it was followed by biochar. A similar trend was also observed when the stress was imposed at maximum tillering as well as panicle initiation stage. But moving on with the irrigated conditions, we could see that similar yield as of that of stressed condition was observed in this potassium recommendation. So from this we can say that silica is actually as good as that when the plant is under the stressed condition as of that it is in the non-irrigated conditions. So this is a cell membrane stability. Membrane stability shows the cell's resistance towards the drought and leakage of its contents. And here we could see that under stressed conditions, highest membrane stability was observed in the silica.
Can you please conclude?
Okay. Moving on with the conclusion, we could see that under the irrigated condition, around 25% of additional potassium recommendation had found a higher yield. Comparatively, the comparable yield was obtained from silica application at the rate of 100 kilograms per hectare from the 50% field conditions. And also here it is to note that under 50% field conditions, application of silica had increased yield around 60% compared to the control without any treatments. So we can go on for recommending the application of silica under droughted conditions to assure the grain yields in rice. Thank you.
Have you worked out the economics of this?
Yes, ma'am.
What is it?
If we are checking on the droughted conditions like 50% FC, we could see that under silica application, we got a highest gross returns from silica application. But under the maximum tillering and panicle initiation stage also, the highest gross returns were obtained from silica. However, comparable gross returns under irrigated conditions, we could get from 25% of additional potash under irrigated conditions.
So it is just gross return, right?
Yes, ma'am. BC also I have worked out, but I just kept.
Which amendment you suggest for drought resistance from your study?
Silica, ma'am. Silica. Actually.
Silica. What about
also
hydrogel? Hydrogel?
Like comparatively, I got silica as the best treatment.
Comparatively, drought resistance imparted by
yes,
silica application.
Silica as followed by biochar.
In that silica application, do you add any additional potassium? Only silica 100 kg per hectare?
No, actually recommended dose of potassium was given as per PAP. Then we have added silica as a
silica only. Then I have a doubt. No moisture stress means flooded condition.
Flooded, yes ma'am, flooded
If field capacity 50% means throughout the crop growth stages.
Yes, throughout the crop.
In that
we worked out the field capacity by seeing that flooded condition. We worked then applied at made it 50%. That then we applied in the first seed.
Okay, so silica application you are suggesting for drought resistance management.
Even biochar we can recommend, but the cost of cultivation incurring is like little higher, so that.
What is the cost of silica in your area?
Actually, it was only rupees 20 per kg when I bought it.
How much?
20 per kg.
20 rupees. It's fine silica only, no?
Yes, ma'am, fine silica, but it differs from
it's
among
not that
countries.
Costly.
Actually, it differs among so many companies and so many sites. It's differing.
Okay, it's not that costly.
Yeah.
Okay, thank you.
Okay, ma'am. Thank you.
Next participant on the dais, OPB03. OPB03.
Good afternoon, respected chairpersons, participants, and those who are present here. Today I am here to present the topic about transcriptome insights into drought tolerance in cocoa for climate-ready agriculture. The main aim is to elucidate the drought-responsive transcriptomic mechanisms in cocoa for developing climate-resilient and sustainable varieties. Especially in Kerala, the cocoa is cultivated as an intercrop under coconut, and we know the recent irregular patterns of the climate changes. So we need to study these mechanisms for a climate-resilient variety development. The main significance of drought in cocoa is a water-loving perennial crop which is high sensitivity to soil moisture deficit, and climate change increases the dry spells in tropics. And drought is a major consequence of climate change. And here it reduces the crop productivity, quality and farmer income, and which alters the biochemical molecular process and the physiological process. And the unexploited research regions include the limited integration of the transcriptomics with field phenotyping, especially the omics integration, and the inadequate functional validation of the differentially expressed genes, and the poor exploitations of the stress memory in perennial crops, and especially the underutilized regional germplasms. And this figure shows, which was shared by the Food and Agricultural Organization about the El Nino impact. In this field, they usually, before days, they usually cultivated the cocoa, but due to the El Nino effect, it intensifies the drought and heat stress in Western African's cocoa belt, and which usually increases the black pod disease and threatening the production, especially it affects the global supply stability. So, in that, the ABA-mediated, abscisic acid-mediated drought-responsive pathway is there. Under field drought, it induces the ABA signaling plus the transcription factors. Through transcriptomic profiling, we can develop the marker development, and hence we can develop the resilient hybrid release. The strategies to exploit these regions is to integrate the transcriptomics with the field phenotyping, especially to incorporate the epigenomics and metabolomics and adopt genomic-assisted breeding pipelines. So integrating the controlled and field studies with the biochemical markers speeds up the selection of drought-resilient cocoa genotypes. So integrating the omics means epigenomics like the stress memory analysis and using metabolomics for the osmoprotectant profiling and marker development for the breeding pipelines. So, usually the cocoa in normal condition, they express the baseline expression under the drought conditions, but it expressed in extensive differential way. So it was different under the drought conditions. So the drought triggers a large-scale transcriptomic reprogramming. In conclusion, it decodes the drought-responsive transcriptomic networks, and we can identify the master regulators for a solution for this. And this transcriptomics enables the precision drought breeding, and region-specific integration enhances the impact. So pathway toward climate ecosystems. So we can decode these genes using the transcriptomics. As a translation impact, it accelerates the climate-resilient cocoa breeding and improves the smallholder farmer income stability and supports sustainable tropical agriculture, and hence it is a model adaptable for other perennial crops. Thank you.
Is this your work? It's just purely a review work you have done. Am I right?
Yes, sir.
So what is your take-home message?
Sir, actually, recently using the transcriptomics mechanisms, they identified 2 or 3 genes in that cocoa, which we can utilize for the further transcriptomics profiling, which stores the stress-related memory of the cocoa. Even after 2 or 3 generations also they are showing. So through this study we can...
So are you going to do this work for your PhD work?
Yes, sir.
So this is just a review of literature of your work, proposed work.
No, actually, a small part is included in, sir, but...
Which part?
That stress memory related responsive part, sir.
That is, you have included that for your technical program. That you have included in your technical program.
Yes, sir, I want to take...
Any of things that you have mentioned here, you have not done the work.
No, sir. Actually, 2 students from here also done the previous part of that work, like the gene expression analysis. From that, they identified the 2 genes, which we can continue and take over the work.
Okay, all the best. Good.
One more question. What is the percentage of drought-affected area under cocoa in Kerala?
Pardon,
What is the percentage of drought affected area in Kerala under cocoa?
Ma'am, about 20% of.
Okay, thank you.
The next participant, OPB04. OPB04.
Good afternoon to one and all present here. I am Devgar Kanandanam, presenting on the topic Building Climate Resilient Genetic Architecture in Pulses Through Induced Mutations. Climate change and environmental fluctuations are one of the major factors affecting the decrease in the yield and altering the maturing and fruiting times, and also increasing the frequency of climatic events like intermittent drought and flood, salinity, alkalinity, which all further on adds to the decreasing the productivity of crops. These factors, coupled with the increasing population, develop a threat to the worldwide food security. Hence, developing crops with which can withstand environmental stresses is crucial for building a sustainable and resilient food system. Pulses are one of the major protein sources for a vegetarian community. Additionally, they are resource efficient crops and improving the soil health, and they can grow under diverse agroecological conditions. One of the major problems faced by the breeders when it comes to the conventional breeding in pulses is restricted genetic diversity, and it can be conquered by using induced mutagenesis and targeting induced local lesions in genomics technology. And through induced mutagenesis, alteration in important traits are done, and which further on increases the genetic diversity. And through tilling, random point mutations are induced throughout the organism's genome, and genomic DNA is pooled and PCR amplified. By creating mismatches between the wild type and mutant DNA strands, we can pinpoint the mutations in the gene of interest. This is one of a case study in which mung bean has been induced, mutagen induced by EMS solution, and further their genomic DNA was isolated, quantified, and pooling and superpooling has been done, and it has been PCR amplified, and the mutation was discovered, and its phenotyping was done. And the deleterious mutations were found for giganti, racemosus, and terminal flowering, and which can be further deployed for delayed flowering, semi-determinate growth habit, and with top pod bearing habitat suitable for high mechanical harvesting. And likewise, some of the varieties which has been used, which has been released are Wamban 12, which has a black gram mutant variety which can be used for irrigated and rice fallow conditions. Trombe Akola Pulse 7 has been released by the Baba Atomic Research Center, which has been a successful variety in Maharashtra due to its early maturity, drought tolerance, and synchronized maturing. And as for now, Kerala does not have any mung bean variety for. And according to the All India Coordinator Research Project, TM96 has been in the ongoing trial for rice fallow cultivation. And on further inculcating molecular breeding, genomic selection, and this can be further made to a core technology in sustainable agriculture, securing the global food supplies.
Are you suggesting future possibilities of doing research in this particular area?
Yes, ma'am.
From review?
Ma'am,
it's a review paper.
No, ma'am. This research has been conducted, but it was not my work.
Okay, so it's a case, from a case you have taken.
Yes, ma'am.
What is mung bean actually? Mung bean, what is mung bean?
Is it green gram or black gram?
Green gram.
Fasciolus mungo.
Green gram only. Previous slide.
Yes, ma'am. Ma'am, Vamban was, I quoted Vamban as a black gram variety.
Vamban is the black gram.
Yeah, yes, ma'am. This TAP7 and TM96 are mung bean varieties. I took the case study in a mung bean variety.
Okay, thank you.
Thank you, ma'am.
Next participant, OPB06.
That was previous one was which number?
OPB04.
Good afternoon, everyone. My name is Haripriya, and I'll be presenting my PG dissertation title, Exploring Bacterial Diversity, Enzyme Potential, and Antibiotic Resistance in Mangrove Sediments of Waipin, Kerala. So moving on. So mangroves are a hotspot for microbial diversity, and they're very important for organic matter decomposition and nutrient availability, and they can be act as potential biofertilizers for climate resilient farming system. So exploring their diversity is very crucial for understanding their specific role in ecosystem functioning. So the objective of the study was to understand the culturable bacterial diversity that is associated with the sediments, to evaluate their enzyme potential and understand the antibiotic resistance associated with this bacteria, and with the taxonomic identification of selected potential bacteria. So the sampling was conducted in 3 particular areas of Waipin, and the bacteria was cultured using soluble marine agar and nutrient agar by using spread plate method and streak plate method, and pure colonies was isolated, and it was subjected to antibiotic sensitivity test and enzyme assay for the following enzymes. So moving to the taxonomic identification of culturable bacterial isolate, as pure cultures was already isolated. From the 5 potential candidates was selected for DNA extraction, and it was amplified by 16S, subjected to 16S PCR, and it was subjected to electrophoresis, and the sequences was obtained by Sanger sequencing, and the database was submitted to EasyBio Cloud to understand the other bacteria that is associated with the particular type, and the phylogenetic tree was constructed to understand the evolutionary relationship. So moving to the results, these are the bacterial colonies that was obtained after plating, that is 26 isolate, and the pure cultures were only 19 isolate that is obtained after quadrant streaking. And enzyme assay result is as follows: 33% of the bacteria shows positive results for the agarase, and 24% for gelatinase, 14% shows positive results for cellulase, and 14% for protease and cellulase, and 5% for catalase, and these are the diagrams for it. And by performing the antibiotic sensitivity test, and I used this diffusion testing, and it shows the susceptibility and resistance of the bacteria towards a particular antibiotics. Moving to the results, out of 13 bacterial isolates studied, 12 isolates were resistant to antibiotic cephalothrin, 11 were resistant to colistin, and 8 were resistant to trimethoprim. For the taxonomic identification of selected bacterial isolates, 16S rRNA gene sequencing was done, and 5 isolates was taken that shows positive for at least 3 enzymes and were resistant to at least 3 antibiotics, and it shows that Cytobacillus firmus shows 99%, 29% similarity. The H1 strain shows 99.29% with Cytobacillus firmus. H2 shows 99.93% similarity with Metabacillus indicus, and H3 with Exigobacterium asterae, and H5 with a 99.86 similarity with Vibrio neocaledonicus. So moving to the discussion part, this shows that these bacteria is highly diverse in producing enzyme, and Cytobacillus firmus and Metabacillus indicus belonging to the class Bacilli is capable of producing certain enzyme that is degrade organic compound, and this Bacilli is known to be explored as bioinoculant, and understanding their antibiotic resistance in environmental isolate help to understand the ecological and agriculture ecosystem, and it will be preventing from pollution. So moving to the summary and conclusion. So these findings highlights the potential of mangrove-derived members as sustainable bioresources for climate-ready agriculture, and the study highlights the different types of varieties and their enzyme-producing capabilities. So moving to the agricultural relevance, the enzyme producing bacteria is capable of degrading the organic matter, and mangroves that tolerate salinity can be used in saline agricultural land. So investigating the role of enzyme producing mangrove bacteria is very important for nutrient cycling. Understanding the genetic mechanism of this antibiotic resistance should be done to prevent the spreading into agriculture environments is essential too. So thank you.
Actually, you have shown similarity to certain bacteria.
Yes, ma'am.
The ones you have identified, right?
Yes,
H1, H2.
Yeah.
So the closest it's saying like 99.2.
Yeah,
So what does it mean? Like, is it very similar? What kind of similarity you are trying to establish?
We cannot completely say that the particular strain is actually Cytobacillus, but it is showing that much similarity, means almost similar traits, and every character that's shown by this particular bacteria is almost similar to this particular Cytobacillus firmus with this much percentage of similarity.
Is there any cutoff for that similarity values? Is there any cutoff values for threshold values for that similarity, percent similarity, to designate it as a new species?
Ma'am, I didn't go it further.
Actually, you are saying like H1, you have identified. So can you say that this is something new? Because already you have close relatives that is saying 99.29 similarity, right? So can we say like this is a new one, or?
Since it is having this much similarity, I think that it will be mostly similar to this cytobacil, the following bacteria.
And the similarity is on what aspects?
I have done the phylogenetic analysis in software. And from that, I've understood that the peculiarities of this by constructing a phylogenetic tree. It shows the similarity of this particular strains to the particular bacteria with this much percentage of similarities by rebooting. Thank you, ma'am.
One question for you. See, you said that most of the bacteria that you have identified, they're showing antibiotic resistance. That means that, what do you mean by that?
Since mangrove ecosystem is one of the most
Yeah, it
productive...
has already been, you're trying to connect that bacterial resistance to antibiotics, to pollution, and you are trying to advise us that we should take enough action to prevent the entry of these bacteria into agriculture system. But the presence of this bacteria itself is, I'm a bit confused.
Since mangrove ecosystem is a productive ecosystem that is meant to produce a bacteria that is capable of producing enzyme that support the particular ecosystem, that is a very functional role. But instead of that, we see an antibiotic resistant gene that indicate pollution and degradation of the particular ecosystem. So, and there is a chance of transferring of this antibiotic resistant gene to other bacteria. Even if enters the food chain, it is causing serious impact, and we humans itself can acquire resistance to antibiotic we are using, and also the beneficial organism, other bacteria in the soil. If that particular resistant gene get transferred to that bacteria, again that may cause problems in agriculture too, degrading the quality of the food that is producing, and from there is a chance of transferring it to food chain and causing a serious
So this is
impact.
a serious study. I think it's a dangerous situation which is being created
Yes.
there. Anyway, you see, this is your MSc work?
Yes, yes. PG dissertation work.
Are you going to continue with this work again? Further studies you are going to take up? Are you going to take up further studies on this one?
Sir, currently I'm doing my bachelor's in education. I hope in future I'll be able to continue.
Okay. Thank you.
Thank you.
Next presentation, OPB07. OPB07.
Good morning, everyone. My name is Harun Zakaria. I'm a MSc graduate from UASB Bangalore. Currently, I'm going to present regarding leveraging microphenomics to improve efficiency and accelerate crop improvement for climate resilience. The climate change has changed the entire stress scenario, the dynamics, and causing an extreme extent where about 20 to 30% of the yield can be devastated in majority cereals, as earlier said, and hence needs hence need an efficient and accelerated way to create climate adaptive, climate resilient varieties. And in order to address this, we need to address the first phenotyping bottlenecks, which are such as observation that could be biased, there can be inter-accessory bias, and usually we assess the endpoint of the disease. And this could clearly cause a bias in the selection. And in order to address this, we need a new tool, a modern tool called microphenomics. In simple terms, microphenomics is nothing but a high throughput automated microscopic image analysis for data collection. This basically gives the intimate details about water use efficiency and helps us understand the disease resistant or disease mechanistic inside the cell and how it causes the defense against a pathogen or how it protects itself. And in order to accelerate or transition from a field-based phenomics level to a microscopic high-throughput level, there are 4 pillars. The first pillar coming to an imaging system, an automated microscoping or fluorescence system that actually visualizes the image without any manual effort. And then a tissue clearing or staining, a standardized chemical process which gives out, can sample out large samples at a fixed period without any deviation from the pipeline. And in an AI pipeline which combines the automation along with the tissue clearing in order to give out extract complex features into measurable data values. These can be integrated into multiple omics such as genomic studies, Jiva studies in order to identify or improve the breeding programs. Before we go forward, how to go with, this is a defining mechanistic microphenotype in order for understanding a defense mechanism against TLB in maize. So basically, I have categorized it into 5 different microphenotypes against TLB disease. One is appraiser success rate, callus deposition, ROS burst magnitude, the growth restriction of fungal colonies, and localized program cell death. So these parameters can be assessed through microscopic models and along with AI, aided by AI, could quantify the disease defense against the pathogen. And using the deep AI or deep learning such as UNet semantic segmentation and YOLO V3 object detection in order to identify and quantify the disease response. And these data could be integrated with Jiva study, RNA transcriptomics, and genomic selection to improve the breeding or research program. How could this impact the crop improvement? The use of microphenomics creates an accuracy and adding large number of samples without any replication. The precision and also can be accelerated by mixing it with other omics for crop improvement such as speed breeding and genomic selection. Thank you.
It's a review paper, no?
No.
How many papers you have gone through to analyze, arrive at the analyzed results?
There is an institute called Leipzig Institute of Plant Science. They are currently working on it. Okay. They haven't published, only one paper in regards to training the model has been published. This is a research proposal with CIMET for my PhD doctoral program.
Okay, have you started it?
No.
Yet to start.
I am planning to go abroad, so this is a proposal that I have planned.
Okay, great. Actually, you have surveyed many papers and you have arrived at your analysis plan. Yes. Okay, great.
Next participant, OPB09. OPB09.
Good afternoon all, respected dignitaries, faculties and participants. I am Amrita Varanya, PhD scholar from Department of Plant Breeding and Genetics. The topic I am discussing today is evaluation of rice genotypes for nitrogen use efficiency, oxidative stress response and root anatomical adaptation under drought stress. So as we all know, rice is a major staple crop and the 2 major factors that affect the rice production is water and nitrogen. So earlier the emphasis was given for high input which later lead to environmental degradation. So as for current scenario, development of a nutrient use efficient rice genotype which is climate resilient is much of an important topic. for sustainable and eco-friendly agriculture. So the objective of my study is to evaluate rice genotypes under combination of nitrogen and drought, also giving emphasis for oxidative stress and root plasticity. So moving to the methodology, the design is 3 factorial CRD with 11 varieties, 2 levels of nitrogen and reproductive stage drought stress. These are the 11 varieties I used in my study. These are the morphological observations that has been taken for screening of the rice genotypes. Moving on to the result, for analysis of variance for these biometrical characters for all the traits, which shows highly significant difference and with 0.01% probability. And by looking at the yield attributes mean value, for grain yield, CO53 and PMK3 showed the highest value. And for number of grains per panicle, PMK3 and Paurnami showed the highest value. And the grain weight per panicle, the CO53 and Paurnami has showed the highest grain weight. And for grain, 1000 grain test weight, Manuratna and Swarnaprabha has the highest value. The nitrogen use efficiency was also calculated, and the variety CO53 showed the highest nitrogen use efficiency in all the treatments, and it's also shown that for the lowest nitrogen treatment has got the highest nitrogen use efficiency compared to the control treatment. So based upon the major outcomes from this initial screening, we could identify 5 best varieties that shows high yield under limited nitrogen condition and drought stress with lower leaf rolling stages. These Paurnami, PMK3, Manuratna, CO53 and Swarnaprabha are the 5 varieties. So the oxidative stress training was also done for the leaf and root of these selected varieties. So NBT staining was done for these varieties for control and treatment, and more dark blue coloration was seen in stressed varieties compared to the control, which shows the accumulation of reactive oxygen species in this variety during stress. This is the pictorial representation in the leaf. Also, DAP staining was done for showing the stress, and the dark brown coloration shows the accumulation of hydrogen peroxide under stress. This is a pictorial representation of DAP staining in leaf. And root histology was also conducted, and the drought stress plan shows an enhanced root modification with increased cell wall thickening and lignification, and also cortical vascular bundle modification, and also air and chyma formation in this root for all the stress conditions. So by concluding my work, by taking this multivariant approach, we can identify the best varieties for developing nitrogen use efficient as well as drought stress tolerant for further breeding program. Thank you.
Which variety you are proposing for the drought stress condition?
CO53 ma'am. CO53 showed the best characters under drought stress among all, followed by PMK3 and Paurnami.
Can you take that slide number 10? You have another slide showing that 1000 seed weight and all those things, no? Yeah. Is this grain weight per panicle, all these things, no? Is it based on the stress condition or normal condition?
All the condition, combination. There are 4 combinations.
Yeah.
Normal condition, that is drought, no drought and full dose of nitrogen. Then next combination is no drought and half dose of nitrogen. The next is drought and half dose of nitrogen and drought and full dose of nitrogen. There are 4 combination treatment, factorial treatment.
And you have taken the average of those.
Value of, mean value of all these treatments. Based upon that only, we are concluded this. And in control, means drought condition also, similar patterns are following.
In normal condition also, similar pattern is covered.
Yeah, normal condition also, there is similar pattern. There is no much difference on par values.
Can you take that slide once more? CO53 is a slender rice, no? Is it bald?
No, slender rice. It's a TNAU variety, ma'am.
And it has a 1000 seed weight of 24.
Yes, ma'am.
And what about Jodi? What about Jodi? Jodi is
Bold rice.
Reported, and even the variety proposals, Jodi's 1000 seed weight is 30.
No, ma'am, this is all treatments, which is control, drought stress condition. It's the mean value of all, not control alone, ma'am. So in Jodi, in drought condition, there is less seed only.
It became slender.
Less seed only.
Less means the more weight is less.
But still, it's a slender variety, no? CO53. It's a slender variety only, no?
Not slender, it's not that much
Small grain size.
Small grain. PMK is much of a slender variety. It's not like basmati rice. CO53 is much bolder.
Okay.
Okay,
Thank you.
Participant OPB 10. OPB 10.
Good afternoon, everyone. Today I am here to present the findings of my research work on the topic screening of rice germplasm for anaerobic germination potential. Anaerobic germination potential, this state, as the name indicates, is the ability of seeds to germinate under oxygen-free conditions. And this state is important in the context of direct seeded rice cultivation that is now emerging as an important climate-smart agriculture technique. In this context, I have selected 250 rice genotypes for screening in order to identify anaerobic germination potential, and these were collected from the Department of Rice, Tamil Nadu Agricultural University, Coimbatore, where I carried out the research work. The design I followed was completely randomized design. Coming to the protocol, seeds were harvested, dried properly, and then sowing was taken up in plastic pots. After sowing, these pots were placed inside cement tanks that were filled with water to a level of 15 centimeter above the pot surface, and this was left undisturbed for 14 days by maintaining this water level. Simultaneously, for the same genotypes, a control condition was also maintained where the pots were left open under aerobic condition for normal growth. In both the cases, 2 replications were maintained for each genotype. After 14 days, observations were taken on anaerobic germination percentage under stress and normal germination percentage under controlled conditions, and these seedling growth parameters were also evaluated. Coming to the results, this is a comparative violin plot. This compares the germination percentage of genotypes under control as well as anaerobic conditions. We can see in control conditions there was almost full germination, like 90 to 100% germination. But in case of anaerobic condition, the germination percentage varied widely from 5% to 90%. This indicates the difference in the response of genotypes towards anaerobic stress condition. Another main trait observed was the difference in the shoot length. We can see in case of control condition, the maximum shoot length attained was just 15.8 centimeter. But in case of anaerobic stress condition, we can see that there was a maximum of 33.7 centimeter shoot length. That is, the tolerant genotype exhibited a very high shoot length, and it was found to be used as an escape mechanism of rice genotypes to overcome this stress condition. This is a correlogram that depicts the relation of anaerobic germination percentage with all the traits that I have observed. It showed a positive correlation with traits like shoot length, root length, anaerobic vigor index, and response index. From my study, a cluster analysis was conducted where 84 genotypes that germinated under the stress condition were grouped into 4 different clusters based on their germination percentage and other morphological characters. It was found that the genotypes in the cluster 2 had the highest values for different traits like anaerobic germination percentage, shoot length, anaerobic vigor index, and response index, which are measured or which are identified as the indicators of anaerobic germination potential. And the genotypes in cluster 4 had the minimum values of these traits, and ideally these 2 clusters had the maximum intercluster distance, which means these 2 genotypes were the most diverse from each other. One set was the tolerant ones and the other ones were the susceptible ones. Also, a genetic diversity analysis was done using already reported SSR markers, and these 3 markers had the highest polymorphic information content and Shannon diversity index, and these were found to distinguish between tolerant and susceptible genotypes. These are the images of genotypes. The tolerant entries were growing out of the water surface, and there I have mentioned the susceptible entry which did not germinate under stress condition, and down it is the tolerant one with good shoot length. From my study, I identified 5 genotypes: RG39, 44, RG41, 7 and 8, and these are land races with good anaerobic germination potential and can be used as donors for crossing program in future works. Thank you.
You worked this in 250 genotypes,
Yes,
right?
ma'am.
And you have given 4 clusters.
Yes, ma'am.
How have you clustered this?
Actually, among the 250, only 84 entries showed germination percentage under stress condition. Those 84 were classified based on the germination percentage. Starting from 5% to 90% was the germination range. And based on the difference in shoot length, root length and all, it was classified based on the seedling characteristics.
How many days you have kept that anaerobic condition?
14 days, ma'am.
14 days.
1-4, 2 weeks.
How many days it takes to germinate?
It started to germinate in, within 5 days, like after 5 days, I started to see that coleoptile elongation.
Still under water.
Yes, ma'am. Not all the entries, the ones that had the potential. Like out of the 250, only 84 showed some kind of germination. All the remaining did not germinate at all till the 2 weeks.
And you selected varieties comes up well also.
Sorry, ma'am, I didn't get.
Selected varieties, no? 5 lines you have selected, no? Whether that will grow.
Yeah, that had more than 80% germination under stress condition. It germinated within the first 2 weeks under anaerobic condition.
And after remaining in water, under water for 14 days, that grows to the full potential.
Yes, ma'am.
Okay, thank you.
Thank you, ma'am.
Then that may be. What was the depth of water?
It was maintained to a level of 15 centimeter above the pot surface.
15, 1.5.
1.5 above the pot surface.
So how many days these germinated seedlings takes to comes above the water?
That from the 10th day onwards, 10th or 11th day, I started to observe the leaves shoot coming out of water surface.
Did you check any submergence tolerance in these varieties?
No, ma'am. These are completely different mechanisms now. Submergence tolerance, like the vegetative growth will be arrested under waterlogged condition, whereas here the shoot was trying to escape out of water.
Okay, thank you.
Thank you, ma'am.
Next presenter, OPC01. OPC01.
Good afternoon all. I am going to present the research study on the topic evaluation of dispensing methods for the release of predatory mite Neocoelus longispinosus against spider mites. In the accelerated phase of climate change, spider mites are being a serious pest of vegetable crops in Kerala in both field as well as protected conditions. The resistance to commonly used acaricides poses a challenge in managing this pest using chemical acaricides. Here comes the need of biological control using predatory mites of Phytosidae family, which has been reported as potential predators of spider mites. Among this, Neocoelus longispinosus is a predominant native obligate predator of spider mites. However, for the successful field implementation, an efficient and user-friendly release technique had to be standardized. With this objective, we have evaluated the efficacy of different dispensing methods to release the predatory mite Neocoelus longispinosus. The experiment was conducted as a factorial CRD experiment under laboratory conditions, which included 2 factors: factor A, predator dispensing method, which includes scrub pad, foam pad, tissue paper roll, and manual release as control treatment, and factor B, which included 3 ratios: predator-prey ratio 1:30, 1:40, and 1:50. For the experiment, mixed stages of Tetranicus truncatus, which is the prey mite, was released on mulberry leaf maintained on wet sponge,
and allowed the prey population to build up for 2 days. And after 2 days, the prey population was recorded. And according to this prey population, predator mites were released using the dispensing methods as shown in the picture at different ratios. And the prey and predator population were recorded at 24-hour intervals. This is the experimental setup. Coming to the result, this table shows the % reduction in spider mite population on 3rd, 6th, 9th, 12th and 15 days after the predator release. On 3rd and 6th day, there is no significant interaction between these two factors, but on 9th day, from 9th day, we can see a significant interaction between the two factors and a 100% reduction in treatment combination of scrub pad and manual release at 1:30 ratio. When we look detailed into the 9th day, which give the highest significant values, the treatment combination of scrub pad as well as manual release of predator at the narrow ratio of 1:30 gave 100% reduction on 9th day itself, followed by foam pad release of predator at 1:30 ratio itself, given 59% reduction, and scrub pad at the ratio of 1:40, given 58% reduction in the spider mite population. When we look on to the individual effect of the factors, scrub pad gave 68% reduction in spider mite population on 9th day, and the ratio 1:30 gave 74% average reduction. So from this, we can say, from the above study, we can conclude that scrub pad can be used as an dispensing method at the ratio of 1:30 for effectively managing the spider mites. And why this method means, it is releasing the predatory mite using a dispensing material is much more easier than the manual release in field conditions. And in future line of study, the field release of predatory mites using this dispensing materials, which is found to be effective, is under progress, and the harvesting, packaging and storage techniques have to be standardized. Thank you.
You have not done this as field release, right? You have done it as a laboratory experiment. You have said you have not done it in the field. You have not released in the field and seen the efficacy of these methods.
Yes, ma'am.
Because when it is coming to the field, the scenario is quite different.
Yes, ma'am. I am a PG second year student. This is my first objective in the laboratory conditions.
Okay.
Now I am going to do the field experiment.
Okay, so the next level is you're going to do
field
it.
experiment.
Okay, all the best.
Thank you, ma'am.
Next participant, OPC02. OPC02.
Good afternoon. I welcome you all to my oral presentation entitled Impact of Gamma Radiations on Egg Parasitoids. Myself Gayatri Kishore, under the able guidance of Dr. Barkat Hussain from Sher-e-Kashmir University of Agricultural Sciences and Technology. So biological control is an essential tool in diverse ecosystems to avoid unwanted effects and impacts on non-target organisms. The egg parasitoid, especially Trichogramma species from Hymenoptera of the family Trichogrammatidae, parasitize the eggs of Lepidopteran pests. Tiny wasps, thus are widely used globally, which control over 400 host species of various insect orders. Trichogramma species have been released in 30+ countries on an estimated area of 80 million acres in diverse ecosystems, including corn, rice, cotton, sugar beet, tomatoes, and vegetables. So the methodology that we followed were different irradiation doses, that is 5 gray, 25 gray, 50 gray, 100 gray, and 200 gray to the adults of Trichogramma pretiosum, and also to the eggs of Corcyra, was carried out in Sher-e-Kashmir Institute of Medical Sciences, Saura, Kashmir. The stage of the insects were the adults of Trichogramma pretiosum and also the eggs of Corcyra species. The layout followed the CRD with 4 applications, including 15 test insects, which was reared up to 3 generations. The machines that was used for the gamma radiations were the gamma cell elite 1, which induced the gamma rays, with an energy of 0.662 mega electron volt and the dose rate at 6.7 Gui per minute. The inoculation of the eggs and the parasitoids onto testing vials was carried out under controlled conditions. The calibration of the machine was carried out. The irradiations were kept using the vials into the gamma radiator. Irradiated eggs of the Coursera was kept for parasitization by the irradiated adults of the Trichogramma pretiosum and finally resulted in the parasitization. The results revealed that there was a strong dose-dependent impact of the gamma radiations in the mortality, survivability, and the parasitization rates of Trichogramma pretiosum. That is, under control condition, the survivability was found to be 100%, while the parasitization rate was found to be 78% with 0% mortality. But with the increase in dose of the gamma radiation, that is from 5 to 200, there was a drastic change in the survivability and all the rest of the parameters. That is, the survivability was found to be 95 in case of 5 gray doses, 86.6 in case of 25, 80 for 50, and finally 1 and almost nil in case of 100 and 200. Similarly, when the parasitization rate was calculated, it showed that it was showing almost 71.15% of parasitization under 5 gray doses. When it was increased to 25, it showed 70, and further, the mortality was resulted in further 100% with 100 and 200 gray doses. The mortality curve was obtained, and the final LD50 values were determined with respect to the time hours, that is 24, 48, and 72 hours, showing a dose-dependent, that is a positive correlation between the doses as well as the mortality was obtained. Similarly, the dose-dependent impact on the hatchability and parasitization rates of the Coursera eggs, that is the host eggs, was also carried out, which showed that with increase in concentration of the gamma radiations, there was a decrease in parasitization rate followed by decrease in hatchability. So to conclude, gamma radiations can be a promising method for pest control, where the impact of gamma radiations on the beneficial parasitoids are less detrimental in compared with the pest. So in case of the Lepidopteran pest, if you have some Lepidopteran host in your field, it reduces the emergence, shortens the longevity, it also impairs the host searching behavior. So irradiated Lepidopteran eggs can be released, which prevents the host hatching. Also, it helps in the storage and handling of the parasitoid development. Thank you.
You have exposed gamma radiation to both the eggs of Coursera as well as to the Trichogramma.
Adults of Trichogramma pretiosum.
What was the practical utility of this work? I didn't understand.
So in order to mass rear the Trichogramma pretiosum, there was a very less production of the Trichogramma pretiosum under laboratory conditions and Kashmir conditions. Once it's winter.
Under Kashmir conditions.
Exactly.
Okay.
So in there, during the field application strategies, so in order to increase the production of the Trichogramma pretiosum, we had to go with the reduced host hatchability. That is, the Coursera eggs was hatching at a very early moment before parasitization. So we had to get a control over the host emergence, for which we had irradiated, and it showed lesser parasitization rate and host emergence when compared to the normal cases.
I'm a bit confused anyway, I don't know. See, Trichogramma pretiosum is an excellent parasitoid for managing FAW.
Yes, sir.
And anyway, you are trying to say that by exposing the host egg,
to
you are trying to prevent hatchability and increase the parasitization. Anyway, I'm a bit confused. I mean, what is the relevance of, I'm sorry, if maybe I'm ignorant, but I don't understand the relevance of this work, a bit confused. You are already exposing your Coursera eggs to UV radiation also, no?
Yes, sir.
And again, you are exposing it to gamma radiation.
UV radiations are basically for the delayed hatchability. But in case of gamma radiations, the results are very much higher compared to the UV radiations. So we are trying to recommend that while mass rearing, that is before releasing the adults onto the field, during the mass rearing procedure, if we irradiate up to 5 to 10 gray doses of radiations, it can reduce the hatchability at a more preventive stages than the UV radiation. So gamma ray...
Kindly check your facts. I'm a bit confused. I'm not sure.
Sir. So it's already published paper.
Published?
Yes, sir.
Okay. Which journal?
Sir, International Journal of Entomology.
Under UV treatment also, we are inactivating the Coursera eggs, no?
Exactly, ma'am. But I'm specifying with respect to the Kashmir conditions. That is, not in Kerala. There, from the October month, the winter starts. So the UV radiations are not being able to produce so much of results. The lab is almost closed for like 1 year. So in that cases, the gamma radiations are becoming a Belton alternative.
Did you compare the both?
Yes, sir. Yeah, I did compare. That was statistical. So, but shortage of work, I didn't include that also.
So instead of UV radiation, you are giving gamma
yes,
radiation?
Sir. Yes, sir.
Okay, thank you.
Next participant, OPC03. OPC03.
What is the number?
OPC03.
Plasma assisted, that one. Title?
What is the title? Transforming.
What is the type?
Actually, the number is not here. C02, after that C04. Which one? Plasma assisted? No, they're not plasma assisted. It is quality index. Transforming the fruit waste into No.
Aparna, can you cite the title?
Here it is. It's
Okay.
not in order. Yeah.
So coming to my topic, transforming fruit waste into functional antibacterial material, a climate smart approach. Myself, Asha MP. Coming to the introduction, we all know that the fruit waste contributes greatly to the environmental pollution. So among these, pomegranate peel, which is rich in antimicrobial phytochemicals, is largely discarded. So this study evaluates the phytochemical content, antibacterial activity of the peel extract, developed a peel-based composite paper, and assessed its antibacterial potential. So the main objective will be fabricating composite paper, analyzing the water absorption and retention capacity, assess the antibacterial activity of the developed paper. So coming to the methodology, I'll just brief it. Going to the extraction, 3 different extraction is being done using 70% methanol, 70% ethanol, and distilled water. The phytochemical screening is being done for 8 different phytochemicals, and this is a test in brief. And the antibacterial assay, agar-well diffusion method is being used, and the test organism was Staphylococcus aureus and Escherichia coli. And the extract volume was 4 different extract volume was used. Paper fabrication, equal volume of the pomegranate peel powder and the waste paper pulp was used, and manual sheet formation and drying is being done. Water absorption and retention test, by using the formula, you can calculate the capacity, absorption capacity as well as retention capacity. And last one, assessing the antibacterial testing of the paper. Again, agar diffusion method is being used, where the composite paper sample 1 into 1 centimeter is being sterilized, and the wet paper disc is being used on testing the antibacterial activity. Coming to the resultant discussion, this is the extraction procedure where the 3 solvent extraction was successfully done, and the variation in the extract color suggests that there is a differential extraction of phytochemical constituents. And this is the phytochemical analysis where the table shows the presence and absence of different phytochemicals, and it is recommended that the alcoholic solvents are more efficient in extracting bioactive compounds compared to the distilled water. And this is the antibacterial activity of the extract. You can see the zone of inhibition, and this is in detail against Staphylococcus aureus. The table shows that ethanolic extract at 15 microliter, it shows a greater inhibition zone, and the bar diagram, it shows the concentration dependent inhibition. And in case of Escherichia coli, the methanolic extract shows a higher zone of inhibition at 15 microliter. Again, the bar diagram, it shows a concentration dependent increase. And next is the development of the peel-based paper. You can see that there is a direct venting of the powder and the pulp, and a stable composite sheet is being developed and demonstrate the practical conversion of the waste into a functional paper material. And this is the paper that is being developed. And next is the absorption and retention test, where 175% is absorption and capacity is retention capacity is 125, and it is seen that there is a high hydrophilic behavior and adequate moisture holding capacity. And this is the effect of antibacterial paper. A high zone of inhibition was seen for Escherichia coli, and there is a broad spectrum of antibacterial activity. And coming to the conclusion, here my study, it highlights the potential of waste, fruit waste into a climate-smart biomaterial. It supports the circular bioeconomy and sustainable material development. With that, I conclude. Thank you.
Actually, you have said that you have dried into powder.
Yeah.
So is there any conditions under which it has to be done? Like how drying has to be done and what are the conditions it has to be maintained for the drying so that you get the maximum composition in it without any loss?
Not direct sunlight. It is being dried under shade. And
how many days? What should be the content, moisture content likewise?
Yeah, it's like it has to be powdered very easily. That will be the condition, and the moisture content should be very, very less. And it takes about
actually, when you're presenting it scientifically, you have to say what is the percentage, to what extent, like how many days.
Yeah,
correct. Exactly. Then only we know like the composition should not change. If you are drying more, sometimes the whole value will be changing, right?
Yeah.
So that was one of the concern.
Yes.
And the second one is you have tested it, the efficacy of it in antibacterial, right? Antibacterial efficacy of it.
Yeah.
So what is the colonies, like how many colonies you started with and how you have shown the suppression?
Yeah, we are swabbing it. It's not that colonies that is being formed. We are swabbing it and then we are placing the paper, 1 centimeter paper in it.
Exactly, but you will be getting the results as number of colonies reduced likewise, no?
No, we are not, we didn't go for the CFU value. We just go for the agar diffusion method only. There is yet work to done.
Okay.
And this is the paper that is being developed.
Okay, how can you relate it to climate smart approach?
Yeah, it is like bioeconomy, that is use, reuse, recycle and regenerate.
Okay, thank you.
Okay, thank you.
Next participant, OPD01. OPD01.
Good evening to all. My name is Jagat Murli and I am an MSc student from College of Climate Change and Environmental Science, Kerala Agriculture University. So my presentation today is on biochar. So specifically, how we can transform Kerala's agriculture waste into a high-value, climate-resilient tool. So in our state, we face a dual crisis, like soils are becoming too acidic and lots of agriculture waste are being built up. So my work proposes a waste to wealth framework to fix both. To understand why biochar is different from ordinary charcoal, we have to look at its structure. Think of it as a biological sponge. So if we look under the microscope, you can see millions of microscopic spores. These spores physically trap water, which is vital during our dry spells. But more importantly, biochar acts as a nutrient magnet because its surface is negatively charged. It grabs essential minerals like potassium and ammonium. This prevents nutrient leaching, ensuring that the fertilizers stays in the root zone where it belongs. So I have gone through various researches, especially in Kerala Agriculture University. And the main focus is on the slow pyrolysis process. And so we take tender coconut husk and woody residues and we bake it at a temperature of around 350 to 500 degrees Celsius. And this isn't just a waste management strategy. It's a thermochemical conversion that locks carbon into a stable form that can stay in the soil for hundreds and thousands of years. So these are the results. Uh, the acidic nature of the soil has gone from 4.5 to 6.5, which is really healthy. And we see a 57% increase in water holding capacity. But for the summit, the most important figure is the carbon math. So every ton of biochar converts like 3 tons of carbon dioxide from the atmosphere. So by doing this, we aren't just improving the soil, we are turning Kerala's farms into active carbon sinks to meet our national climate goals. So how can we implement this? So we don't need expensive like factories or industrial plants. We just need to use low-cost kilns, which can be used by smallholders, and this can be produced in the site itself. This framework reduces the fertilizer dependency and improves the food security as well. By turning our local waste into permanent carbon lock, we are building a resilient Kerala for the next generation. Thank you.
It's a review paper. Have you reviewed the?
Yes, I have gone through
Okay,
various...
what is the basic pyrolysis temperature for biochar production?
It ranges between 350 to 500 degrees Celsius.
350 to... Do we have any recommendation of biochar for any of the crops in Kerala?
Specifically, I can't say one right now, but there are various researches happening.
Can you say the recommendation for rice?
It can be used in both ways for reducing the acidic nature of the soil.
I asked the recommendation dosage. Do you
Dosage.
How many tons?
I don't know.
How many tons?
No, I don't know.
Actually, you have shown that pH has been increased from 4.5 to 6.5.
Yes.
In that study, what was the recommendation followed?
I didn't look into that.
Okay. You can go through and you can find it.
Okay, ma'am. Thank you.
Next presenter is OPD02.
A pleasant good afternoon to my respected dignitaries and my dear research scholars. Myself, Sadisti, from Department of Agriculture, Soil Science and Agricultural Chemistry, from College of Agriculture, Vellani. And today I am here to present a part of my research study entitled as Assessment of Carbon Stock in Major Land Use System in Southern Laterites of Kerala. Why did we choose this study? To set the stage, there are approximately 800 billion tons of carbon in our atmosphere, but a staggering 2,500 billion tons of carbon are stored right beneath our feet. The topography of Kerala is like due to heavy rainfall, almost all nutrients and organic carbon were leached out. Yes, the study has been, the objective of the study is to assess the soil carbon stock in the major land use system of Southern Laterites of Kerala. Our study area was AU8, Southern Laterites of Kerala. From this AU, 4 different land use systems were selected. Soil samples were collected from the surface and subsurface. From each land use system, 20 samples were collected and processed for further analysis. Total 80 samples were collected. Basic soil physicochemical properties were assessed like pH and EC, and bulk density by core sampler method, and organic carbon by black radiation method, and soil carbon stock by Ratan Lal's formula. And then the four land use systems were banana, coconut, and rubber, and current fallow. The results were the soil pH, the mean pH of the fallow land was recorded as highest mean pH values, whereas rubber land use system recorded as lowest pH, whereas soil EC, both surface and subsurface fallow land use system recorded as lowest, whereas highest banana EC was recorded in surface, but coconut recorded as the highest subsurface EC. The mean bulk density value of banana in surface recorded as the highest bulk density, whereas rubber land use system recorded as the lowest in both surface and subsurface. The total soil organic carbon percentage, where mean value is recorded as rubber perennial cropping system as the highest, whereas fallow land use system as recorded as the lowest. The mean carbon stock was assessed, and then banana is recorded as a surface highest carbon stack, whereas rubber contributes to the highest subsurface carbon stack. Conclusion was the pH decreases with the increase in depth, same for bulk density, EC and organic carbon also, whereas uh, the carbon stack decreases with an increase in depth across all land use system. The highest surface carbon stack was found in the banana land use system, whereas the highest subsurface carbon stack was found in rubber soils. Thank you.
How many sites you have selected for sampling?
Total 80 sites.
Total?
40 sites.
40 sites.
Yes, ma'am.
For all the areas you have calculated the carbon stack up to 45 centimeter.
No, 30 centimeter.
30 centimeter.
0 to 15
so 15
surface.
and 15 to 30.
Yes, ma'am.
Based on up to that depth only you have calculated.
Yes, ma'am.
Okay. Can you justify why rubber is having higher subsurface stock and banana is having?
Due to high litter fall from rubber, and then it fasten up the acidification process, ma'am. Due to acidification process, the microbes cannot decompose the litter.
Okay, surface stock is more in banana and subsurface stock is more in rubber. Can you justify the?
Like the litter has been converted into decompose very quickly in banana, ma'am. But whereas in rubber, the litter decomposition process took a little while.
However,
and then bulk density also higher in banana, whereas rubber contributes while less bulk density due to its undisturbed soils.
Okay. Okay, thank you.
Thank you.
Next presenter is OPD05. Please note, next presenter is OPD05. Next presenter is OPD06. OPD06. Next theme, OPE01. Judges, please note, next theme, OPE01.
കയറ്റിയായിരുന്നു ഇവിടെ വന്നിട്ട് PPT ചെയ്തിട്ടില്ലേ? ഓക്കെ. ഇത് ഇന്നലെ PPT ഉണ്ടല്ലോ. PPT അയച്ച് നേരത്തെ വന്ന് കയറ്റാൻ പറഞ്ഞായിരുന്നു. ഗ്രൂപ്പിലുണ്ടായിരുന്നു. ഇല്ല അല്ലേ? ഇതാണോ സാറേ?
Good afternoon, everyone. My name is Anuranj PR, and I'll be discussing on the topic, understanding the drivers of awareness and adoption of climate smart technologies and practices in coffee production systems of Kerala. As we all know that climate smart agriculture is a set of agriculture practices which helps in boosting productivity, enhancing resilience, and reducing greenhouse gases. And also, FAO have cited that CSA is context specific in nature. Climate change significantly reduces global coffee production suitability 50%. Coffee is a highly climate-sensitive perennial crop, leaving less chance for the coffee farmers to suddenly rotate the crop. CSA supports long-term sustainability of coffee landscape. As we all know, studying coffee is also important because it is second largest consumed beverage in the world and the second largest exported commodity just after the crude oil. And the research gap, we found that there was no integrated and weighed adoption existing, and the prior studies focused mainly on individual practices, and also there was no expert prioritized CSATP inventories present. The objective was to assess the level of adoption, to estimate the adoption levels, and to identify the determinants of awareness and adoption. We chose Wayanad, Palakkad, and Idukki districts. With the help of the data from the Coffee Board, we selected Muppayanad Mepadi from Wayanad, Nelliyambati from Palakkad, Elappara and Vandiperiyar from Idukki, and sample size of 175 was formed with the help picking 35 farmers randomly from each of these panchayats. Uniqueness of the production systems was also analyzed to see or to study the adoption pattern. For that, we didn't find any inventory of CSADP, and the first contribution from our study is the contribution of the inventory of CSADP. We followed literature review, expert stakeholder consultation, and sorting. We made it into 4 dimensions: soil management practices, water management system, as well as the crop management practices. We sorted it out to 20. We did an analytical hierarchy process to weigh with the criteria of technical support requirement, cost effectiveness, subsidized nature, less labor intensive, and resource conserving. And we finally found the weights of each procedure or each practices and technologies which we use. We have 2 benefits from this. We got the first ever inventory of climate smart practices in coffee, and we weighed the system, weighed the procedures which we used in the adoption levels. And also we assessed the level of awareness with this inventory, and also the level of adoption was seen using Mirati et al.'s adoption index score. And one way ANOVA revealed that there was moderate awareness and regional significance or variation was very less in water. And conceptual framework was we used to analyze determinants using a framework called Political Ecology Framework by Madden, and also probability regression models was used for analysis. We use this framework because the like CSATP adoption can be resulting from more than individual choices, and they influenced by access to resources, power dynamics, and environmental constraints. We took variables from socio-economic sphere, institutional political sphere, ecological systemic sphere, and water management. We ran the bivariate and multivariate probit analysis, and these are the significant findings that we have made, and we imply that the adoption is shaped by socio-economic, institutional, and ecological factors. Policies must better align subsidies and incentives, and technology adoption is not just technical, and it requires socio-ecological understanding. And extension must go beyond technical advice and address behavioral and social barriers. Strengthening farmers group is a key, and adoption depends on land, labor, and local context. Promoting low-cost, locally suitable practices with strong institutional support is important, and addressing constraints before promoting these technologies is important, and designing region-specific strategies for better scaling. These are the institutional linkages observed during a visit to all these areas of high ranges. I conclude by saying that strengthening climate resilience in coffee systems requires not only awareness, but also context-specific support that enables farmers to act. Thank you.
Okay, thank you. And you have told about inventory. You have prepared an inventory for this. What is the process you have, the procedure you have followed for it?
Madam, we have gone for an literature review first.
Okay, that I got it.
The second one is expert and stakeholder consultation. We have visited Coffee Board, each and every offices in these 3 districts of Wayanad, Idukki, and Nelliyambati, and also the progressive farmers, which includes in the Coffee Board, and the license offices also was visited. We listed out the practices. We sorted it out with the help of the Deputy Director of Research and Joint Director of Extension from the Coffee Board. Then we analyzed and listed out practices and sorted it into 4 dimensions and 5 practices under each dimension, making it a 20 practices.
How many experts you have checked actually for this?
5 experts from Wayanad, Idukki, and 3 experts from Nelliyambati, considerably based on the availability and the area under coffee production and the stakeholder and the extension organization or institution present in that specific area.
Okay. Why couldn't you add more experts who were working on this line? Why? Because the more, it will be more better.
Madam, I have also validated with the analytical hierarchy process, which states that it is, I know you know analytical, it is a multi-criterion decision process. This was also with 4 experts, according to the SATI, the father of this analytical hierarchy process. 1 expert is very much enough to run the AHP process. We have validated using 4 experts, and also the geometric mean was done and normalized to weigh the process. And we first consulted 5 experts, and we had to drop 1 because of the consistency ratio. And we used 4 experts, which only 1 was required. And with these weights, we finally, and all these 4 were in alliance, and it complemented each other, and it stayed together. So we finalized the inventory of 20 practices.
Okay, and political ecological framework, you have quoted somebody's work on that. Have you
Madam,
modified it based on our local situations, or you have used it as such?
The framework usually say that every components, rather than, local conditions was seen. I have went 3 times for the survey to all the high ranges, and at that time, I interviewed each and every progressive farmers also. And at that time, we could understand that the field conditions were different too. So based on, ma'am, can you repeat again the question?
No, actually I was asking political ecological framework used by somebody you have adopted.
Yeah.
But I am asking you the conditions, local conditions, whether you have considered before framing it. That was my question.
Yes, ma'am. Local condition was because in the first preliminary study and the first round of study, we went to Wayanad Coffee Growers Association and all, and the organic certification systems also was followed there. We also found institutional backup and organization backup also.
Okay, thank you.
So we modified accordingly.
Okay.
Thank you so much.
Next participant, OPE02. OPE02.
Yeah, I'm Hari Guru Prasad, PG scholar from Tamil Nadu Agriculture University. I am going to present about the assessment of digital climate readiness among the small household farmers in Tamil Nadu. If we see, means in Tamil Nadu, in all the state in India, mostly 85% of the farmers are small and marginal holder farmers. So, how the farmers are adapting the digital tech like climate smart agriculture and agri tech, how they access the digital technology, like there is a problem in that adapting the adoption rate. So I had taken this as the research gap and doing the work. Most of the farmers have, after the pandemic, farmers had the access to the smartphones, but some farmers had engaged the advisory practices, but not following the adoption practices. So here I had constructed the index called Digital Climate Readiness Index for the small household farmers, and I had taken the 2 districts and compare the what is the DCRA index ratio and justify the result. Here I have used that study area. I have selected 2 different zones, western and southern agroclimatic zone. In that 120 farmers are selected. They are the small and marginal farmers from the 2 districts. 60 farmers from each district I have selected for analysis. Here you see that the DCRI components and the frameworks are, I had listed this. The DCR, the Digital Climate Readiness Index, which was framed, constructed to assess the, how the farmers is preparedness to the utilize the digital information for adapting the decision making. In that, we had constructed this DCRI for assessing how the adaptive practices followed by the farmers. So, in which we had constructed from inspired from the 2 frameworks, indicator frameworks from the previous literature work. One is from the climate readiness and the adaptive capability literature. Here I have listed the components. You can see that the 4 majorly I have taken the 4 components: digital access, digital capability, and advisory engagement and adaptive practices. In that each component I have taken the different separate indicators. Like in that digital access, we can see that whether the farmers having the smartphone ownership and internet access, whether access, and in digital capability we can see that WhatsApp usage, availability and forecasting. In advisory engagement, we can see that frequency of checking updates and the digital group participation. In participation, whether the farmers is opting the digital information, whether the list out the information as the farmers is practicing in the real in their farm in this field. This is the methodology I have used. I have listed the component and the indicator I have selected. Then I have scaling. I have normalized it and I had followed the aggregation technique, and then I had given the DCRA scores. Here the results you can see about the comparing with the two districts, Erode and Theni. Here we can see the 4 components like a digital access and digital capability and digital engagement and adaptive practices score. The total DCR score is for Erode 0.61 and Theni 0.53. Here there is the slightly different compared to the both districts. But if you see that adoption rate means it is low. The farmers has the digital access high, and compared to the digital access, the digital capability is low, and the final most the adaptive practices is compared to the it's half of the adaptive practices they had followed. These are the DCRA category I have listed, and the categories according to the farmers using the adoption practices. This is the policy and policy suggestions I had given that extension who are the farmers contacted with the extension faculties and extension officer. They had using the digital practices, the digital tech to improve the yield and all efficiency. Here I had inferred that concluded that the digital infrastructure, which means the farmers who are adapting the digital has increasing its ability to adapt.
Okay, how have you operationalized adaptive practice? What is the operationalization?
Yeah, if you see means in the website, we have developed the agri-tech digital platform like forecasting weather and weather apps. So they had listing the weather forecasting in that whether the farmers is adapting the forecasting rate. Whether we showing that the sowing date and the cultural practices that are the time period, whether the farmers are adapting this time period to improve the efficiency or else not. Like that I had mentioned the adaptive practice.
So you have said access is more, capability is less, which means like they have all these things but they are not utilizing it.
Yes, ma'am.
Is it what you have inferring on that?
Yeah.
Right. And when it is coming to adaptive practice, it is still more.
No, ma'am.
Still less, sorry, still less.
Yes, ma'am.
Okay. So which means that access is there, but when it is coming to adapting the practice, adopting the practice, it is still more lesser.
Yes.
Right.
So
what are the reasons for that? Any reasons suggested for that?
They are accessing that digital platform and engaging, but they are not coming forward to adopt that adaptive practices. So the extension people or extension of a person can conduct the training on digital literacy and adoption rate to increase that adaptive practices.
So actually Tamil Nadu Agriculture University is giving these details, right? The information for them.
Yes, ma'am.
Is it that, or you are in toto you have checked?
Total I have.
It is not TNAU recommendations advisory alone.
Yes, ma'am. Calculated total apps.
Is it TNAU advisories, or is it like general agricultural aspects?
General agricultural aspects.
General, everything, every app, everything related
yes,
to agriculture.
Yes, ma'am. I had studied and included the other aspect, not only the TNAU portal aspect, and the other apps also they are listing.
Whichever apps, okay. And you have got which apps they are using more frequent, like which apps they are using for getting information other than TNAU advisory, and what is the extent of usage of TNAU advisories for this?
That are the other objectives, and
there are other objectives that you have to do.
Yes, ma'am. According to time,
fine. Okay. You are yet doing it?
Yes, ma'am. Currently working on it.
You are continuing it?
Yes, ma'am.
Okay, fine.
Next presenter is OPE03. OPE03.
Good afternoon. My name is Vishnu. I am a master student from A3 Bangalore. My research is titled Drivers of Farming System Change and Intergenerational Adaptation to Climate Change in the Cold Wetlands of Kerala. This study explores how rice-based farming in a unique wetland ecosystem is changing over time, and how different generations of farmers perceive and respond to these changes. So my study is located in the coal wetlands in Thrissur, which is one of the India's major agro wetlands, rice production system and a Ramsar site. This region is heavily dependent on seasonal hydrology, collective farming institutions and pump-based water management, making it extremely sensitive to climate resilient variability. Coming on to the problem context, recent years show increasing climate variability, erratic rainfall, flood, heat stroke, shortage of labor, input delays, which are affecting the farming system timeline. Traditional crop calendar are shifting. Profitability is uncertain. And younger generations are gradually moving away from farming. However, these changes are rarely studied together in an integrated way. Coming on to the objective and research gap, this research aims to understand how farming system and crop calendar have changed, identify the climatic and socio-economical drivers, and also examine how different generations perceive and adapt to these changes. There is a limited systemic research linking climate change, labor, mechanization, and intergenerational adaptation in the wet-dry system, which the study addresses. Coming on to the methodology, the study uses a mixed method approach, combining the farmer survey, focus group discussion, key informant interviews with institutions, farmers, farmer organization, and researchers. Data were collected across both the older and younger farmers, pump operators, Padashakara Samadhi leaders, and the policy actors. Also, the baseline variables from the CARA project are integrated with the qualitative narrative with the farmers to analyze the farming system change. As the work is continuing, some of the key insights are suggesting multiple drivers of change, including the climate variability, labor shortage, increasing mechanization, institutional constraints. Seed availability is the major concern, and with the continued dependency on the older varieties like Uma and Jyoti, and increasing the pest pressures here. Generational differences are clear here. Younger farmers emphasize more on the economic viability and technology, while the older farmers are more focusing on the environmental uncertainty here. Coming on to the contribution at the next step, this research contributes to understanding the adaptation limits in wetland dry system by linking the ecological change with institutional and generational perspective. Next step, I'm going to include the analysis of the crop calendar, economic viability, and the adaptation pathways across the generation to inform the policy on seed systems, labor support, and the climate resilient wetland agriculture. So these are some of the references I used. Thank you.
Can you just show the methodology used?
Yeah.
Okay. You have actually conducted all these baseline household surveys, semi-structured interviews, focus group discussions. I think there should be a data set for all these things.
Yeah.
When it is coming to we get a set of data, right? And if you have presented it with the data and analyzed the data, I think it would have been more factual.
Okay. Actually, it's the end of the data collection. I haven't finished the data collection. That's why I haven't included the analysis part.
Understood. But definitely, I think it's a huge data that is going to be generated from this.
Yeah.
And when you are analyzing, it will be a very good paper.
Yeah, sure. Thank you.
Next team, Team F, presenter OPF01.
Good afternoon, dignitaries, fellow researchers, and my dear friends. Today I stand with a question. How do you gonna, that sits in the heart of a climate smart agriculture, how do you gonna reduce the climate footprint with the human's oldest crop, that is rice? So, still rice sustains more than 3.5 billion people worldwide. In Kerala, 2.3 lakh hectares support food security and rural livelihood despite land use pressures. Flattered rice promotes around 8 to 12% of the global anthropogenic methane emissions, with having the global warming potential of 27 to 30 times. And India's rice cultivation emits nearly 3.97 teragram of methane annually. Despite Kerala is having the contrasting rice ecosystem like flood-prone, below mean silver like Kuttanad, irrigated monsoon-driven dual season Palakkad especially, spatially explicit block level methane emission maps are lacking, limiting the accurate regional greenhouse gas accounting and target climate smart intervention. So my research gonna address this. The study area includes the Kerala, that is Palakkad and the Kuttana, which are the major rice growing tracts of Kerala. So with an objective of development of an integrated model for the quantification of methane emissions from paddy fields using the geospatial data and to estimate the methane emissions on a regional scale. This is the methodology. We use the SAR data, this is synthetic aperture radar. We use Sentinel-2 optical images and Sentinel-1 SAR images. We undergone the pre-processing with the help of NDVA and the VS backscatter. We have quantified the cropping season, identified the rice area mapping, which include the start of season, peak of vegetative stage, and end of season. For the ground truthing, I used the GPS-based field location mappings. I surveyed the areas at a block level. Like we have calculated phenological stages, photography, planting data, harvest, organic amendment, fertilizer factors. Survey covered Kuttanad 15 blocks under the Punja season and the Palakkad 5 blocks, Virupa and Mundakan. The quantification of methane emissions was carried out under the IPCC Tier 1 techniques, which undergo the 2019 refinement, which uses the default emission factor and the region-specific scaling factors like water, pre-season flooding, organic amendments. With this, we're going to get that adjusted daily emission factor. If we multiply that adjusted daily emission factor with the duration of the crop and the area of the crop, and multiply with the 10 to the power of -6, we're going to get the gigagram of methane emission per block per year. So validation, we have used the static closed chamber technique, which is a golden rule in the methane emissions, and we have model performance assessment with the help of the percentage deviation. So we have gone for the block level methane emission calculation framework. So I have already published a paper that is for the rice area delineation using the Sentinel-2. So we have mapped the Kuttanad and the Palakkad, which accounts for the 46,961 hectares in the Kuttanad and the 20,520 in the Palakkad area, with having the good more than 90% of accuracy, with the more than very good agreement of the 0.87 and 0.83 of copper coefficient, with the blocks of 15 blocks in Kuttanad and the 5 blocks in Palakkad. So this is the classified 5 land use and land cover for the both Kuttanad and the Palakkad, depicting the paddy areas. So next we use it for the duration, which is used for the estimation the methane emission. We have used the Sentinel-2 NDVA and the Sentinel-VS backscatter, which observes the measure. The dominant variety is Uma variety, which is having the standardized duration of 120 days. We have gone for the farmer survey, concerned with agriculture's for department officials and the field survey observations. So the baseline emission factor, this is the baseline emission factor we have find out, which is actually multiplied with the soil water regime scaling factor, pre-season flooding factor, and the organic amendment scaling factors. This is the water management practices and the pre-season flooding organic amendment practices, which are derived for the both Kuttanad and the Palakkad, depending upon the farmer surveys and the department officials. And we have we have find out that adjusted daily emission factor for all the 15 blocks and the 5 blocks, and the annual block emission for all the 20 blocks. So I have given an example how we're going to estimate this. Like the baseline emission factor is 0.85 with the water management practices of 0.71, 2.41 and this. And we have got the annual block emission for the Velian block. So this is the annual block emission for the Kuttanad. And this is the map depicting the spatial distribution and emission intensity. And similarly for the Palakkad district. And this is for this. And for going for the validation purpose, we have used the static closed chamber technique and estimated samples in the 4 stages. And samples are analyzed in Tamil Nadu for the Uma variety. And we found that the percentage deviation from the IPCC is actually much lesser, showing the less. Even though Virupa is showing the much deviation, but it's under the IPCC acceptable range only. So this is conclusion like this. This is the first block level methane inventory for the Kerala. It's a high accuracy spatial maps are there, actionable regional insights, which is actually scalable scientific framework, which is ready to adaptable to the diverse tropical rice growing areas. Thank you. And if anyone want to contact us, they can scan and connect. So there you can find our papers. And we are looking for the collaboration also.
How did you do the ground truthing of the data by measuring CH4?
Yes, ma'am. We have taken the static chamber and we have analyzed
using
static chamber technique.
Estimated using GC.
GC, ma'am. Gas chromatography.
And what is the schedule? Have you analyzed throughout the crop season?
Yes, ma'am. From in the 4 critical stages like tillering, panicle initiation, flowering, grain filling, and the maturity stage.
Only during the critical stages you have assessed the methane emission.
Yes, ma'am.
And
we have
from those values you have calculated to the total
duration of each stage, and then we will get the seasonal methane emission for the entire duration.
Is it the standard protocol?
Yes, ma'am. It is the standard protocol by the GSD protocol of international.
Okay. We used to see weekly sampling.
Yes, ma'am.
This is only critical stages.
Yes, ma'am.
During critical stages, there will be this paddy is under flooding.
Yes, ma'am.
Completely
I
flooded?
have just replicated the former field practices. I have taken the samples from the Kuttanad and the Palakkad districts. So I have taken the data from the
And
farmer field districts.
according to your paper, which area is having more methane emission?
Kuttanad is having the more methane
What
because
is the
since
reason for that? Can you?
Kerala is having Kuttanad is having around 45,000 air rice hectare, and it is having the pre-season flooding factor that during the there is only one cropping per year, ma'am. They will go for a long flooding.
Per hectare emission is more in Kuttanad.
Yes, ma'am.
Okay. The reason is?
Reason is since there is a pre-season flooding factor, ma'am, the land will be flooded for nearly 8 months. There will be only one crop per year.
Okay. Does it have any relation to soil carbon or something like that?
Ma'am? Yes, ma'am. And Kuttanad area is having the higher carbon stock.
Okay. You told that a scale, the values are below the acceptable
yes,
rate. What is the actual
according
value?
to IPCC, it is around the 15 to 30% range.
Okay. Okay. Thank you.
And the values are acceptable. It is not a model failure. It is just because of the monsoon hydrology that's actually observed in the Virupu season, ma'am.
Actually, what is the critical level that should be treated as cautious? Do you have any?
Since it is from agriculture, there is no critical level. Actually, it is found out that how much effect it cannot create, how much methane can emit. There is not actually standard value that below which there is a critical. So there are some European areas they can produce more than 600 kg per hectare, ma'am. So our values are still lesser.
But do we have any scaling?
There is no particular, I have gone through the literature, there is no particular ceiling for the methane from the rice area.
Okay, thank you. However, it is with respect to the critical stage values only, not the entire crop cycle, it's not monitored, no?
According to the international guidelines, the methane samples has to be taken in the critical stages itself.
Okay.
And since, like in say, it's MSc work, so it's a shortage of...
Okay, thank you. Good work.
Okay, ma'am. Thank you, ma'am. Thank you so much.
Next presenter is OPF03. OPF03.
Good afternoon. Myself Shaling Christ, MSc first year, Department of Agriculture and Meteorology, Kerala Agriculture University. I am here to present my study, Evaporation Paradox in a Warming Climate, Seasonal Trends in Reference Evapotranspiration using Penman-Monteith equation. Reference evapotranspiration represents evaporative demand of the atmosphere, and it plays an important role in water resource management as well as drought and climate impact assessment. So for this, long-term variability and seasonal trends in evapotranspiration, climatic variables are collected from 1985 to 2024, that is 39 years of weather variable data are collected. From this weather variable data, reference evaporation trend can be estimated. The study area is conducted in Department of Agriculture and Meteorology, Kerala Agriculture University. So, the weather variables include maximum temperature, minimum temperature, relative humidity, wind speed and sunshine duration. These weather variables are collected from 1985 to 2024. From these weather variables, reference evapotranspiration can be carried out by Penman-Monteith equation. From this reference evapotranspiration, trend analysis can be carried out by Mann-Kendall trend and trend slope estimation. From this trend analysis, sensitive analysis as well as the contribution percentage can be estimated. So this monthly distribution of reference evapotranspiration, we can see in pre-monsoon period, the reference evapotranspiration will be maximum. And when it comes to the monsoon period, it shows a decreasing reference evapotranspiration and it increasing towards the winter. So this shows a clear seasonal cycle with decreasing reference evapotranspiration from summer to monsoon and increasing again towards winter. In season-wise trends reference evapotranspiration, we can see in winter pre-monsoon, southwest monsoon and northeast monsoon, we can see the slightly decreasing trend in all 4 seasons from 1985 to 2024. And same-wise increasing trend is followed in relative humidity and same as in sunshine duration. For maximum and minimum temperature, we can say there is a significant increasing in the trend. When it comes to wind speed, it shows the decreasing trend. So comparing the sensitivity analysis and the contribution percentage, in winter season, the contribution percentage is high in wind variable, and pre-monsoon it is high in sunshine duration, and in southwest monsoon it is high in the sunshine hours, and in northeast monsoon we can say relative humidity as well as sunshine duration show highest contribution percentage of 39.5 and 39.4. From this season-wise sensitivity, contribution percentage, and season-wise trends, we can say that decreasing trend over time in every season is mainly due to wind speed and sunshine duration, and this behavior confirms the evaporation paradox. That is, if temperature increases, evaporation decreases, and this confirms the evaporation paradox. This study is useful for improving irrigation planning and crop water management. Thank you.
What is your conclusion?
Ma'am, from these weather variables, we can say that the reference evapotranspiration will be higher. And this reference evapotranspiration, we can conclude that...
It's based on which data you have studied? How much?
These are weather variable data, ma'am.
Weather. Okay, how many year data?
Yes, ma'am.
30 year.
Yes, ma'am. 39 years.
Okay, this is one. Okay. And your suggestion is? Okay. Okay.
Thank you, ma'am.
Next presenter, OPF02. OPF02.
Dignitaries on the dais, faculty members and fellow researchers, good evening to all. And I am here to present on the topic, identification of high-performing tree species in the multifunctional agroforestry system, a field study at the early stage. So the objective of the study was to design and develop a multifunctional agroforestry system, particular to the northeastern zone of Tamil Nadu. And this research has been taken place at Valavachanur, one of the sub-district where the Agriculture College and Research Institute, Valavachanur, has been placed, which is under TNAU. And we all may be familiar with the topic of agroforestry. Agroforestry is incorporation of single tree crop in an agriculture field. So when it comes with the multifunctional agroforestry, it is the incorporation of different agroforestry model in a single cropping area. So this is the field design, an experimental site. A total of 8x8 has been taken as land size, and it has been separated into 4 quadrants, each quadrant specific to each agroforestry model, like quadrant 1 for tree-based agroforestry model, and quadrant 2 for fruit-based agroforestry model, and quadrant 3 for timber-based, and quadrant 4 for high-value tree-based agroforestry model. A quadrant spacing of 4x4 was maintained for each quadrant, and a total of 18 species of trees has been selected. And number of trees or the tree samplings has been planted is 5. So I have computed the volume data of different tree species, and from this data it is evident that the tree species like Syzygium cumini and Pithecilium dulis and Tectona grandis, which 2 of these are fruit-based agroforestry model crops, and they have a very high growth performance during the early stages of the life. And here you could just see that some of the value has been computed as 0, which suggested that those crops didn't grow well in that area. And one of the key factor to be taken into account is that this study has been taken place or been carried out in a degraded land. So obviously in a degraded land it would not suggest some of the species which require of high nutrient values. And I have also computed the carbon sequestration level, and we all know the carbon sequestration level is directly correlated with the volume of the tree species. And this is the graphical representation of the data. Here, the data which has been computed didn't follow the normality, so other statistical analysis has been taken place, and here, as a statistical ranking, it is also giving the same results as highest median for Pithecia diluis and Syzygium cumini following Tectona grandis. Here, I have just integrated whole volume at the 90 days after planting, and the representative carbon sequestration level of the tree species has been documented here. And some of the limitation is like the period of study was so low, so the benefit cost ratio towards the farming society can't be incorporated into the study, and it has been taken place in a single location only. And I thought of taking this opportunity to create an awareness on the multifunctional agroforestry system. And special thanks to Dr. S. Manikam sir and Dr. S. Vanilla ma'am, Dr. V. Arun Kumar sir and other fellow students who have helped me to carry out this project.
Okay, thank you. How have you selected this combination?
It is based on the service of the trees, ma'am.
Like you are saying like fruit-based agroforestry model, Syzygium cumini, Annona squamosa. How have you selected this? Any criteria for that?
Yes, ma'am. The service provided by those trees, ma'am. Like Syzygium cumini is jamun and Annona is custard apple, or the fruit-based or grouped into a fruit-based agroforestry model. And similarly, the tree species which provides seeds of high oil content has been grouped into tree-borne oil seeds group, and the timber-based or the trees which is primarily used for the timber purpose, and high-value tree species or the similar of timber-based, but the revenue generated from the high-value tree species are pretty higher.
And you have projected your data till 90 days, right? Of
Yes,
volume.
Actually, it was one of my part of my UG work, so within that period of
It's
4
continuing.
months,
Because the tree canopy, when it is growing, it will be a different condition
Yes,
altogether.
ma'am. That's what I have just mentioned in the limitation also. But I thought of exploiting this topic here for the creating an awareness on multifunctional agroforestry system can also be included, as this system, apart from the service to the nature, it also generates an additional income to the farmer as different components. Even if one of the component gets failure, or if the crop which has been which has been selected by the farmer fails, we can generate an additional income from the other parameters or other
Can we
factors.
do a modeling or something regarding this, like what can be the canopy, like maybe futuristic conditions?
Sure, ma'am, I'll see.
Yeah, you can just, if you can project that, it will be well and good. Better than this, much better than this.
Yes, ma'am.
Do you have any replicated locations or a single location?
No, ma'am. It was conducted in a single location only, ma'am. But similar thing was also carried out in FCRI Metupalayam. And based on those recommendations and...
Are you following that also? Are you analyzing that system also? Only this one?
That is a different objective, ma'am. I have separated it for it. And for this presentation, I have given this one location alone.
Okay, thank you.
One question.
Yes,
You have listed 5 tree species as well as 5 fruit-based trees, 5 different trees,
Yes,
plants in
yes.
each agro ecosystem, no?
Yes, ma'am.
So, and number of plants, trees planted as 5 also.
5.
Did you conducted this experiment?
Yes, ma'am. We have just conducted it, ma'am. As you can see, that is the actual area of a plant.
Is it in the same field these 5 trees are planted, or in
In the
different...
same quadrant, ma'am.
Same quadrant?
Quadrant.
So, there will be 25 plants, tree species. In the
5
first-
plants of each
in the
species.
first- that means total 5 plants of each
Total
means
of 25 individual tea crops will be in one quadrant.
That is in 4 by 4 meter space.
Yes, ma'am. The density would be more. As here the land selected is Thank you all.
Presenter OPF04. Last and final presenter OPF04.
Good evening, respected Chair and distinguished participants. I am presenting our research title, Multi-Scale Spatio-Temporal Drought Assessment in Humid Tropical Kerala under Climate Changing Conditions using SPI and SPEI. The focus of this research is to understand how drought behaves across different agroclimatic zones of Kerala under changing climatic conditions. So, usually, the drought studies is mainly focused by the precipitation-based indices like the SPI. But in these climate changing conditions, the indices which measure the evapotranspiration also need to be assessed. By this, we formulated this objective to examine the temporal variability, severity, and spatial heterogeneity of drought conditions across agroclimatic zones of Kerala using multi-scale SPI and SPEI under climate changing conditions. The study area was, for this study, we subdivided Kerala based on different agroclimatic zones like the central zone, high rain zone, northern zone, problematic zone, and southern zone. And the study period was from 1983 to 2024, and the different weather variables we used were the monthly maximum temperature, monthly minimum temperature, and precipitation data. And the drought analysis was done using the SPI and SPI for 3 different time scales, like the 3 months, 6 months, and 12 months. And the trend analysis was done using Mann-Kendley test and sense slope estimation. And the drought was characterized based on frequency, duration, intensity, and severity. And for understanding the correlation between SPI and SPI, the Pearson correlation test was conducted. Now let's go into the result and discussion. First, the temporal variation of SPI and SPI at different time scales. At the 3-month time scale, all agroclimatic zones exhibited high-frequency wet-dry oscillation, indicating rapid surplus and deficit conditions. Importantly, SPEI3 constantly showed stronger negative anomalies compared to SPI3. This indicated that short-term drought intensity is amplified by atmospheric evaporation demand rather than precipitation amount. When coming into the 6-month scale or the seasonal scale, the variability becomes smoother compared to the 3-month scale, indicating greater drought persistence. SPI6 indicated that higher drought incidence than SPI6 across zones, suggesting that temperature influence became more evident at seasonal scales. In the case of 12-month scale, prolonged dry and wet phases were clearly captured, and the significant negative departures of the rainfall were observed during 2000 to 2005 and 2012 to 2016. And the northern and high rain zone exhibited a strong long-term drought signals, particularly under SPI 12 rather than SPI 12. This highlights spatial heterogeneity in long-term drought exposure across Kerala. When the trend analysis was performed using the Mann-Kendley trend analysis test, and for all agroclimatic zones and for all timelines. In southern zone, that both SPI and SPI across all the time scales showed an increasing trend or wetting trend, and with a small census value. In the case of central zone, the drying and wetting condition highly fluctuating, so it showed no trend, but Z value remains positive. In case of northern zone and high rain zone, the 12-month scale showed the increasing trend for both SPI and SPI. In case of problematic zone, there was no trend, but the Z value was negative, showing a drying condition. And the drought characteristics were done using 4 different parameters. In the case, first one is the duration. In case of drought duration, based on the heat map, we can see there is a higher drought duration 12-month scale, mainly in high rain zone for both SPI and SPI. In frequency, also the 3-month scale showed the highest variation than the SPI. In the case of intensity, the SPI showed higher variation compared to the SPEI. In the severity, the hilly rain zone and the problematic zones showed higher variability in the SPI, and in the case of SPEI, high rain zone and northern zone, highest drought severity. A strong Pearson correlation between SPI and SPA across all zones and timescales confirm that precipitation remains the primary driver of drought variability. However, SPA detected a higher intensity, frequency, and severity of drought events. This demonstrates that amplifying role of atmospheric evaporative demand under warming conditions. We can conclude in this changing climatic conditions, although precipitation remains the primary driver of drought, the atmospheric evaporation demand also have an important role. For the effective monitoring, we need to integrate precipitation and temperature-based indices. These are my references. Thank you.
what we have observed, some of you are taking 1983 as a start point of your observations, and it goes up to 2022. Then some people are taking 1983 up to 2023. Some people are taking 1983 up to 2024. What was the logic behind this thing?
Sir, actually, when I'm going to conduct the study, when I check for the database from 1983.
So it means that your database starts only 1983.
Yes, sir. For the correct, for my data, the precipitation data was the.
Not only yours, we have seen many previous speakers also similar work. They are already all of you are starting your base data with 1983 year. So we are a bit curious. What is the relevance of 1983?
So actually,
it means that either you have the data with you
yes,
from
sir.
1983.
Yes, sir. We are having data from the 1983.
You have taken data from 83 to
2024, ma'am.
That is 41 years.
Yes, ma'am. 41 years.
42 years.
41 years.
42 years. Is there any significance for this 42 years?
Ma'am?
Is there any significance in your study with this 42 years data?
No, ma'am. Actually, due to this global warming or the changing climate, the rainfall pattern is shifting from year to year. So I think we can, if we include the maximum number of years to back historical data, we can get a much better result. That's why I have used the maximum data which is possible to, that is available.
Okay. Is it the data available from 1983 onwards only here?
Yes, ma'am. Our department was having that data from the precipitation data from that year onward.
Okay, okay. Okay, thank you.
Thank you.
That's clear our doubt.
The oral presentation has come to an end. Next, we move on to the prize distribution ceremony of the Student Summit. First of all, we have the results of poster presentation. I invite Dr. Binupi Boni, Director of Extension, to give away the prizes. I also invite Dr. A. Leda, Director of Student Welfare KU and Convener of Student Summit, to give away the prizes. The results of poster presentation. The first prize goes to chess number PPC12 S. Saki Priyanka, Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Puducherry. The winners will receive memento, certificate of appreciation and cash prize sponsored by IRI Kera. Second prize goes to chess number PPC02 Amino Sulhat TA, Kerala Agriculture University. Third prize goes to PPC01 Santosh Babu R, Tamil Nadu Agriculture University. Thank you, ma'am. The results of oral presentation will be announced soon. We extend our sincere gratitude to everyone who made the Student Summit 2026 a remarkable success. Within just 2.5 weeks, we received an overwhelming response with participation from 25 institutes across South India, including 35 oral and 47 poster presentations in Theme 1 and 25 oral and 10 poster presentations in Theme 2. Our heartfelt thanks to the South Regional Agriculture Fair 2026 team for organizing this event at Kerala Agriculture University, and to the Government of India and Government of Kerala for their generous sponsorship. Special appreciation goes to convener and organizing committee of the Student Summit, the dedicated supporting staff and enthusiastic student volunteers whose hard work made everything run smoothly. Yeah. The results of oral presentation is here. First prize goes to Nandana AS. Chess number OPB 10, Nandana AS, Tamil Nadu Agriculture University. Second prize goes to chess number OPC 02, Gayatri Kishore, Kerala Agriculture University. Third prize goes to OBP 06, Haripriya, St. Joseph College of Ernakulam. St. Joseph College of Teachers Education, Ernakulam. Thank you, ma'am. A big thanks to the judges of today. Dr. Veena Vigneshwaran, Associate Professor, RRS Vaitila. Dr. Gavas, Associate Professor, DRS Kannara. Dr. Tulasi V, Professor, RARS Pattambi. Dr. Smita KP, Assistant Professor, KVK Thrissur. A very big thanks to all the participants. A big thank you to all young researchers whose vibrant participation brought energy and innovation to the summit. We also acknowledge the KERA team for their cash prize and continued support. Thank you one and all.